Yeah. That's
Okay.
No, let me get some water. What's the snow melt? But it was real, I was getting snow on the way. I think it's supposed to be.
No, that's not like.
I'm seriously watching that like.
I know, my back here.
Is this yours, Natalie?
Yeah. Yeah.
Yeah.
Really.
A whole hour? All right, everyone, welcome back to those of you online. I think we are ready to get started with our afternoon session. I'll turn it over to Hannah, who's going to be moderating this for us.
All right, I hope everyone can hear me. I guess for a little background, we usually as a group talk about what topics we'd like to learn about or every spring, and we talk about that in the fall. So we basically decided the topic for this spring was going to be integrated weed management in conjunction with biocontrol. But if you come up with ideas that you'd be interested in, always present them to Melissa and we will talk about them and try and identify some speakers for the future. So to get started, we're going to have our first presenter is Natalie West, and she's going to be presenting on the general considerations for integrating biocontrol with other tools. Natalie is with the U.S. Department of Agriculture, ARS, which is Agricultural Research Station or Service in Signey, Montana.
Yep. Great, thank you. I'm going to keep this brief because there's going to be a lot of people talking after this that have a lot more experience than that. My role is a lot of weed ecology and biological control. And so I'm going to just give you a brief overview today about how I think about integrating biocontrol with other tools. And biocontrol, as we know, can be a very effective technology, but it was never really, it's never been considered a technology to be used in isolation. Much like chemical really should be conducted in combination with other management tools. Biocontrol can be most effective when it's integrated. So I started this off with a little picture up here of my technician setting up a weather station, because this thing has a bunch of different things that do sensors that tell us all kinds of different things towards one common goal. So we all know that biocontrol is exploiting sort of ecological interactions to control weeds. So, and our management tools emulate these ecological stressors. So all these insects, right, are just really putting some pressure on the plants so they're not going to survive. And we talk a lot about biocontrol here and we think it's valuable, but whether insect herbivory generally has a controlling influence on plant populations is a testy discussion in plant population ecology. There are wars over this in scientific discussions. But when biocontrol is successful, it can be a cost-effective and long-term solution to weed issues. But success really depends on proper deployment and combination of tools. Because if you think about it, insects, right, they evolved to eat plants, but if they're going to eat themselves out of house and home, then that's not a very good evolutionary strategy. So what we need is sometimes some extra tools to really help biocontrol to be most effective. So impact, I don't always equal control. I think that's a big thing to consider, right? And here's because the fact is like you can have a lot of insect feeding, but maybe you're not controlling your plants. Why is that? As I said, co-involved organisms have learned to deal with one another, right? They're in a constant arms struggle. Plants can respond to and defend themselves against herbivory, right? They have all kinds of strategies. And we don't always know what the most vulnerable stage is. A lot of these plants evolved in another range. They came here, and maybe all the little interactions and different things that they've had may make them actually vulnerable at different stages than they were in their original range. And the thing always to remember is many plants that are weeds here were also weeds elsewhere. So it's not a new thing. Sometimes it is new. Sometimes they really are. These interactions that are making them weedy, but sometimes plants are weedy wherever they were. And so all these things we need to consider when we're thinking about weed management programs. And so we have these variations in success results from a lot of different influences through the landscape. So maybe our insects aren't very, have these sort of habitat limitations where there are all these different aspects. Weeds, right? They can control, you may have mismatches in phenology or compensatory regrowth that defend them against insects. And then there may be all these kinds of things feeding into the plant community. So I think the thing to think about, and I know we've focused on a few different areas here to talk about in weed management. In integrating your management, the most important question is, what is the management goal? And so people today are going to talk a little bit about different things that have influenced different strategies that could be combined with biocontrol. But the most important thing to consider is, we're talking through this, is like, how are these things most useful to you in reaching the goal that you've set for yourself? So what is your management goal? Depends on what weeds you're working on, right? And what kind of the characteristics of that weeds. It depends on the land that you're managing. What other management is happening? What other tools were less successful? What are you trying to help bolster? Do you want to eliminate patches or stop spread? Are you trying to stop these things from getting bigger, or are you trying to stop weeds from really taking hold? What types of agents are available? Sometimes we can't control that, but we want to be cognizant when we're thinking about how we want to manage these weeds, what types of agents are available to us. And finally, really, what the management goal is, what does success look like to you? Sometimes biocontrol isn't going to get rid of all of your weeds. How many of your weeds are acceptable to you? Or what does your plant community need to look like? So when we're talking about integrated management, really we're talking about multiple stressors. When weeds are attacked from multiple directions, they have a less chance of response recovery. And so that's pretty good for biocontrol, right? And in general, for weed management, reduces resistant selection, increases our long-term chance of effective chemicals. So, and it also reduces the possibility, thinking about integrated management reduces the possibility that control just open spaces for secondary invaders. One recent paper suggested that at least 25% of controlled invasions just results in re-invasions or secondary invasions. So, you got to think about what you want to come behind your weed management. There's weeds coming around behind you. So, in general, what are the concerns for biocontrol efficacy? Timing is crucial. These insects have their own sort of phenology. And so your management techniques need to not limit your insect success. And so thinking about how much food is available to the insect might be part of that. If you spray your weeds at the wrong time, but you want your adults to eat enough to lay enough eggs, then you're going to want to think about not spraying all your plants out. Agents disperse. So you don't know always that the agents that you have are actually able to your patch. Are they coming out of your patch, or they're dispersing from elsewhere? Is there an actual limitation where things are not seeming to control, or is it an actual lack of control? Sometimes that's the question. Disturbance frequency. I always say weeds like disturbance, but insects like some predictability. They need to know their food source is going to be there, so you want to think about that. And there's a possibility that some introductions will always be doomed. And sadly, there's been a lot of discussion today about the lack of post-release monitoring and our understanding. And this is really a big question, I think, a lot of times. What actually limits insects in the environment? We don't always have consistent information on that. And so we don't know whether introductions are doomed or whether repeated introductions will eventually establish populations. And also, weed reduction reduces insect habitat. So you want to think about the fact that as your weeds decline, your insect habitat will decline. And so thinking about keeping populations going through time is going to always require that possibility. The other thing I always, as a plant ecologist, want to say is establishment doesn't always equal control. Because you have agents doesn't mean that those agents are going to have an impact. It doesn't say they won't over time, but it might be 10 years after you establish them. So thinking about that and how long you need to be tailoring your integrated management efforts towards supporting your insect population is going to be important. So before we release, you want to think about how you're going to manage your insects, how your weed and your insect management, weed kind of phenology and insect needs are going to interact with the management that you're imposing. And also, I always want to put out a call to remind you to document your. Like I said, today we've talked, like it's come up multiple times, how important it is to have the monitoring. And so when we're establishing integrated management programs, part of it is understanding what we've done and what has worked, hasn't worked. So those are just some introductory things. And I will leave it to the next person, unless you have questions.
Right, thanks, Natalie. Are there any questions in the chat? Okay, if there's no questions in the chat, our next presenter is Carrie Mintier. They're with the University of Florida, and they're going to be presenting on the review of research on integrated weed management incorporating biocontrol.
You should be able to share. Oh, looks like you can.
Right. Are you guys seeing my notes screen or the actual?
We are seeing the slides.
Okay, so
And
not the notes.
No, just the slides. Yeah.
Okay. Yeah, this is my first time with Microsoft Teams, so my apologies on this. Okay, so hello, I'm happy to be here, at least virtually with you today. So Natalie's presentation was amazing and is gonna integrate directly into mine, pun intended, very well. So so I'm gonna get into a little bit more specifics about some of the research that's been done on integrating biological control with other weed management techniques. So you all may have seen this already today. I just popped in, so you might see it after me. This is a pretty important figure in invasion biology. This is the invasion curve, right? So this shows us how invasions look like over time. So where we start off with some very small number of localized populations right after introduction. So we're in the eradication phase. Then we move over into the containment phase, where our weeds or other invasive species are spreading, but we're still trying to contain them into like smaller areas. And then you get into this really nasty, angry red version of the curve, right? And so this is where we're just looking at resource protection and long-term management. Now, we can control invasive weeds in a number of ways, and you all are very familiar with all of the different ways that we can control them. What tactic we're using is really indicative kind of where we are in this curve. So in the eradication phase, we're using chemicals, we're using mechanical controls, we're using fire. We're really trying to get these things out of here, right? Like kill them, get rid of them at all cost. Containment, you're still using a lot of these. You might be using some cultural controls at this point as well. But when you get into like resource protection and long-term management, that is when my phone usually rings, right? Like, Carrie, we need biocontrol for this particular species. We have so much area infested that we need something that's going to be effective on a landscape level, right? Because it's hard to get out there with enough chemicals. It's hard to get out there with enough chainsaws, right? In order to be able to make an actual impact when you're talking about landscape level weeds. And so, so that's kind of when biocontrol usually comes in. Now, biological control, as Natalie said in her presentation, isn't always meant to be kind of like the panacea, right? Like, you're going to release an agent, Carrie, and then we're not going to have any more of these weeds. And she covered that really well on like why this isn't the case, right? Like, classical biological control is not going to eradicate a species. It's just going to lessen the density of these species on the landscape if we're lucky. Now, we have been lucky several times. So these are some of the insect species in Florida that have done really, really well for us, at least in Florida. So this is like the alligator weed flea beetle, the tropical soda apple leaf beetle, and the air potato leaf beetle. These insects did a tremendous job with reducing the density of these weeds on the Florida landscape. Most of these don't do as well outside of Florida because of winter. Some of these insects don't really like freezing temperatures. And so if the weed can survive, but the insect can't, then of course you don't get a whole lot of control. But in Florida, these guys have done pretty well for us all by themselves. There's been lots of research done on this, of course. The research shows that it's estimated between 3 and about 30% of biological control of weed agents. So these are specific biological control species are effective. So that's 3 to 30% of the species that we have introduced have been effective. Now, based on definitions by Hoffman, where complete control is no other control is necessary, about 25% of biological control projects have provided complete control. So that is not species, like biological control agent species, that is projects. So like spotted knapweed out there in Montana, we've had 13 agents, different species of agents released on this plant. So that would be a project, right? Like you're looking at all of those different insect species. So about 25% of the time, the biological control can actually provide complete control where no other control methods are necessary. That doesn't mean the weed is gone. It is still there, but it is at a more tolerable level, right? The literature also shows that about 50 to 75% of biological control projects provide substantial control. So this is control that we're happy with. We are seeing impact. However, other control methods are necessary. And so a majority of these projects, you do need other control methods to be able to get these plants where we want them, right? So biological control can be viewed maybe not as a panacea, maybe not as a silver bullet, but as a very useful tool in the weed control toolbox. And this is especially true for trees, evergreen and perennial vines, and some fast-growing aquatics in nutrient-rich environments. And so these are some of the examples that we have here in Florida, where we have had to throw integrated management at these targets. So this is Melaleuca, Brazilian pepper tree, water hyacinth, and old world climbing fern. So integrated pest management is needed, right? We've seen this. A majority of our biocontrol projects also need some other weed control methods. So you can often get synergy, right? Like you can get increased control when you use a combination of tactics than either control tactic alone. There are, of course, special considerations with biological control agents, and I'll cover these a little bit later in the presentation. Call for integration. Biological control researchers and invasive plant, people working with invasive plant control have been calling for integration with biological control for multiple decades at this point. This started back in the early 90s. I was not the first one to suggest, yes, we should really be doing this more often, and I'm not going to be the last. But so there's a list of authors that have already done this, and I'll talk about a paper that me and my colleague Ellen Lake put together to see if we've actually been doing this more often, because people have been asking for this for quite some time. Okay, so this is the paper that me and my colleague Ellen Lake put together. It was published back in 2018. And what we did is we did a review of the literature over 30 years. So from 1987 to 2017. And what we did is we went to Google Scholar. We used the search terms integrated management and biological control or biocontrol. And then we added that in with the Latin name of a target plant. We searched all weed species in Rachel Winston's 2014 catalog on the biological control of weeds. So this is a lot of weed species. It did take us a lot of time. This book is phenomenal, and I use it almost every day in my work. But so we did this with every single weed species in the Winston et al. book. And we searched through 3 pages of results. So that's about 30 references for each to check them for suitability. Past the 3rd page, you are getting things that are distantly related, and we weren't interested in those. So how did we qualify these? So we saw these 3 pages, right? This QR code will actually link you to that paper if you are interested in looking at the references or looking at the more detailed methods for this. So we were interested in field or common garden studies. We were interested in studies that had classical biological control combined with another tactic. We concentrated weeds that were problematic in natural areas or rangelands, so we stayed out of agro ecosystems. We concentrated on peer-reviewed literature only, and then we analyzed the trends in what we saw. And what we found is we got 39 studies that were concentrated in terrestrial ecosystems and 16 studies that were in aquatic ecosystems. Now, this is very likely to have underestimated the number of studies that are actually out there. Number one, our search terms. If you would have changed those search terms, even tweaked them a little bit, we probably would have gotten a couple extra studies. We also concentrated on only peer-reviewed studies. These are things that are in the peer-reviewed literature. Many projects looking at integrating biocontrol with other weed management techniques are done by land managers, landowners, and they might be published in newspapers, trade journals, things other than peer-reviewed studies. We were not going to be able to look at all of those. So this is strictly in peer-reviewed journals, but this is what we found. So this is not that many over a 30-year time period. And what we found is we ran some statistics looking at number of papers over time, and we found that there was no increase in number of studies reported annually in the peer-reviewed journals. Most common weed targets were spotted knapweed, so that's 20% of the papers we found were in that system, and leafy spurge was about 16.4%. So it's very clear that you all out west do a much better job at this than we do out here in Florida. After this talk, you'll hear a talk from Dr. Kelly Carruthers about some of the work we have done in Florida, but currently you guys are doing a much better job with 20% and 16.4% of the integrated studies in peer-reviewed journals. Okay, the tactics that are most frequently used with biological control are herbicide at 36.4% of the studies used herbicide. 23.6% of the studies used plant competition alongside biological control. Bottom of the list, so used the fewest in the number of studies was grazing at 7.2% and mechanical control at 3.6%. We also found that using more than 2 tactics alongside biocontrol, or 2 or more tactics alongside biocontrol, was about a little over 16% of the studies that we saw. All right, special considerations. So what I'm going to cover now are kind of general considerations. And then I'm going to discuss special considerations by weed management type. So special considerations, biological control agents are living things, which means that if you kill them, they don't provide any impact on your plant species, right? So you have to partition your other control tactics either in space or time to not harm or eliminate your biological control agents. This is of the utmost importance. You can do this by providing refuges because these plants or these insects, remember, are host specific. So if you completely kill their host, they will not survive. You can spray, cut, burn when agents are not active so that you're not going to be directly damaging or harming these insects by your control tactic. You can also select control methods or products that are not harmful to your control or to your biological control agents. And Kelly will talk a little bit about this in the next talk. This, unfortunately, is different for each and every system, every insect, every weed, and very likely specific habitat types as well. So we're going to move on to the more specific things that are just for each particular weed management tactic. So we're going to start with herbicides. From the literature, and all the literature is cited down at the bottom that I pulled these from, is that the presence of biological control agents can actually reduce herbicide concentration needed, exposure time, or frequency of application. So this can actually reduce the amount of chemicals that you're having to use. That saves money, and that's more environmentally friendly. Herbicides can, unfortunately, alter the population dynamics and or kill agents. They can do this directly by the insect being sprayed with them and then being penetrating through the cuticle, the chemical penetrating through the cuticle, or by the insect ingesting of sprayed plant material. Can also do indirectly by elimination of the food source. Remember, these are host-specific organisms. It's also shown in the literature that impact of a chemical on agents is reduced when you have slow-acting herbicides. And this is because oftentimes immatures that are occurring on these plants can actually complete development before that plant dies and then disperse into a better location where the refuges that you'll leave, right, are. And so slow-acting herbicides can actually be a little bit more compatible with living biocontrol agents. Moving on to plant competition. Plants attacked by biological control agents can often be more susceptible to plant competition. And so that's great, right? That's literally what biological control is supposed to be, is reinstilling that natural balance, right? So our native plants are having to defend themselves against the insects that are feeding on them. And now we're requiring the invasive plant to do the same thing. We're leveling that playing field, right? So it makes them more susceptible to competition. Plant competition has also been shown to reduce the invasive treadmill. So the invasive treadmill is when you control one invasive, another invasive comes in its place. So if you have a nice, healthy ecosystem with not like a lot of native plants, you can reduce this occurrence of this invasive treadmill. Unfortunately, oftentimes desirable species may not be present, especially in degraded landscapes. So degraded rangelands can be extremely problematic with this. And so passive restoration is unlikely, right? If you don't have the propagules for your native species there, you don't have enough native species to be able to fill in these gaps as your invasives die off. Passive restoration is unlikely. Unfortunately, planting of natives can often reduce survival of the biological control agents. And so you really have to figure these out on the system basis. Moving on to mechanical control. Mechanical control, of course, removes old growth and senesced foliage. This can actually stimulate new shoots, which can be great. Because these shoots can sometimes be more palatable to biological control agents. They can sometimes be less palatable. So I feel like my ecology professor from college just that always told you two things, right? It could be this or the exact opposite. And so it's something you really have to figure out for your system. New growth on these can also be better able to uptake herbicides. So mechanical herbicide and biological control can sometimes be a really good combination. Damage can also make the target more susceptible to phytopathogens. So if you're using like a fungal biological control agent, these work really well together to get more bang for your buck as well. And timing. Natalie talked about timing a couple times in hers, and I will talk about timing a couple times before the end of my presentation. Mowing is important to time right. If you have insects that are developing inside your inflorescences of your plants and you cut them down too early, your insects are going to die. You don't want to take out your next generation of biocontrol agents. All right, moving on to fire. Patchy burns can be good. I know, the horror. Like we don't want a patchy burn, Carrie. It can actually be great, right? So you'll have refugia for your biocontrol agents, and this can facilitate dispersal of agents into other areas, right? We'll keep some of your agents there for when these weeds make a resurgence. Fire frequency, you have to pay attention to this. You have to give the agent time to recover to damaging levels. If you burn too frequently and you're cutting back on your biocontrol agent population, you're never going to get them up to the level where they need to be able to really help you out. Timing again, you need to time your burns when the target weed is most susceptible. You need to time your burns when the agent is least susceptible. And you need to time your burn so that weed regrowth is most vulnerable to competition. So a lot of moving parts here. Additional challenges, small scale studies are useful. So this is the first step, right? Like common garden studies, things in the lab. However, we need larger field studies to demonstrate the applicability of this to land managers. I've never met a land manager that says, yes, Carrie, I will completely change my management because you have this little lab study, right? We need larger field studies to be able to demonstrate this applicability. You have to be able to maintain the treatment for sufficient time. So this is long-term to monitor plant community changes. This can be particularly challenging on multi-use sites. But to wrap up, IPM and biocontrol, I think we can be besties. We can reduce pesticide use, which is great, extend time in between treatments. We can get greater control, reduce the invasive treadmill effect, but it has to be investigated for each system individually, and that's the hard part. Thank you for your attention. Here's my contact information if you want to contact me and ask any further questions, but I think I might have a minute or two for questions.
Are there any questions for Carrie? Any online? All right, thank you, Carrie.
Thank you.
Next up, we have Kelly Carruthers from the University of Georgia, presenting on the integrating biocontrol and herbicide.
Okay, hang on, let me figure out how to share. I'll just share my screen and make it weird. I'm sorry. Maybe. Okay, there's a thing in the way. All right. Let's see. Swap. Which one are you seeing, the regular screen?
You're seeing the correct screen, yep.
Okay. The magic of online presenting. And so what screen are we seeing? Well, thank you guys so much for having me. I'm going to kind of talk specifically about some of the things that Dr. Mentiro talked about of integrating management using a biocontrol agent and chemical control specifically for Brazilian pepper tree, which is a woody stemmed plant. So it could be potentially useful for some of these kind of large tree issues that we also see. Okay, there we go. So Brazilian pepper tree, just to give you some background, it was introduced in the late 1800s into Florida. It is an ornamental and it is beautiful, but it is aggressive. So it's a part of the family Anacardiaceae. It's really lovely in that capacity, but it's also related to poison ivy and will do just what poison ivy does and cause contact dermatitis in people who are allergic. So it has very similar properties and problems, which hopefully you don't experience with salt cedar or something else like that that you may have issues with, but it's particularly problematic in Florida because of that. It produces numerous bright red berries and they're easily dispersed by birds. They love to feed on them and then spread them, and they end up in a variety of ecosystems, including our native mangrove forests in Florida and the Everglades. But then it will also disrupt and degrade habitats for threatened and endangered species like the gopher tortoise. It's, classic invasive species. It's doing all the harm we can think of. It's particularly problematic through much of South Florida. It's problematic in California, and then every island of Hawaii also has presence of Brazilian pepper tree. So those three states are kind of the major states that have problematic Brazilian pepper tree, but we do find records of it in Texas, Alabama, and Georgia as well. And with the prospect of kind of global climate changes, it's not likely to decrease in spread. It will spread along these kind of coastal regions through the southern U.S. So it is not likely to just disappear on its own. It's likely to spread. And that's where we need kind of more management strategies, because mechanical and chemical control are happening, but it's costly and it's inefficient. There's over 283,000 hectares of Florida that are covered in Brazilian pepper tree. And despite the efforts to, bulldoze it as we see here or spray it, it's still dominating the landscape in a lot of areas. So it did lead to the development of a biological control program. And in June of 2019, we released Pseudophilothrip zucchini, a thrips, which feeds on kind of these newer shoots of the tree. And so we're hopeful that it will get there, but the idea is that if we can kind of integrate an approach with using the biocontrol agent and then the chemical controls that are already used, we might actually be able to reduce some of the herbicide usage, but either maintain or increase control efficiency for this plant. And so we're going to start kind of with direct toxicity studies. That's kind of the foundation layer for what we did. And as Carrie so beautifully mentioned, there's kind of two different scales that we'd have. So we have these small scale experiments that we started with. The direct toxicity is a lab study, and then an immigration study, which is a screenhouse study. And then we scaled up to the field studies. And although I'd like to show you all the graphs and all the data from what we did, because I'm very obnoxiously proud of it, I will kind of try to hit the high points so you can at least kind of get the idea of what we did, and then feel free to reach out if you have any questions about how we did it. So starting with direct toxicity and immigration, these are the main chemical control components that we knew could work alongside biocontrol. There are other ones, but they'll reduce the foliage completely, and we need to keep the foliage there, has been mentioned several times, as we need a food source. And so to do that, there's two ways. We can use a foliar application of an herbicide, which sprays the whole foliage to complete coverage, but not to runoff. And for Brazilian pepper tree, it's typical glyphosate, imazapyr, and triclopyr amine are used. And then there's the basal bark application. So the lower 30 centimeters of the bark of the tree is sprayed with the herbicide. And in this case, we really only have triclopyr ester that's used most often by land managers. So those are the chemical controls that we started with to try to form this kind of integration project. And so the basic objectives of the studies was to determine direct and indirect toxicity of herbicides commonly used for Brazilian pepper tree management to Pseudophilothrip zucchini. And then we wanted to know if Pseudophilothrip zucchini would immigrate from our herbicide-treated Brazilian pepper tree to an untreated plant. And so to kind of just hit the high points of direct toxicity, when we put thrips in a Petri dish, just like this with some filter paper, we'd put 5 thrips in there, and then we would spray them directly with an herbicide, just directly, and then seal it up. And over the course of 5 days, we really had high survival rates regardless of what herbicide we sprayed on them. Over 71% of the thrips survived the course of 5 days being sprayed just basically right in the face with an herbicide. For the foliar application, we ended up applying the herbicide to the actual foliage of the plant and then fed that to the thrips. And we found that imazapyr was the least harmful. As Carrie mentioned, this was one of those slow-acting herbicides, so it gives us a lot of time for some healthy tissue, and it didn't seem to bother the thrips over the course of 5 days. We'll talk about the triclopyramine in just a second, because we also did the same with the basal bark. We applied triclopyr ester to the base of the trees and then fed that leaf tissue to the thrips, and they did not like it. So we had a survival rate of about 51%. And I kind of air quote that one because anytime that we used triclopyr, our thrips went missing. So if I started with 5 in the container, I might end up with 3 by the end and then try to figure out among 3 who was alive. So it was a tricky thing. And if you worked with thrips, you know they're crafty and they can get out of just about any container. So I was calling them Pseudophilothrip Houdini by the end because I was very frustrated with their disappearing acts. But that's how it goes. So we went using that information, we went to move towards an immigration study. So we used our kind of least harmful herbicides. We did the imazapyr, and we compared that to a water control just to see the movement of thrips. We used kind of the concentration that's label rate for spot treatment. So this is a fairly high concentration to actually be used in the field. We also included methylated seed oil, the adjuvant, which the adjuvant is just there to help make sure that the herbicide makes it into the plant and can effectively do its job about killing the plant. And so we compared that to water. And then we used triclopyr ester, our only available basal bark treatment. And again, higher concentrations for that spot treatment with the adjuvant of basal bark oil there. We also did bark oil on its own just to kind of get an idea about how those two would compare. And so what that would look like in this study is that we would find two plants and pair them about the same size, label them treated and untreated. And then our treated plant, we would apply a treatment, in this case water, and then waited 24 hours. After that, we paired them in the cage. Into the treatment plant, we gave them about 35 thrips, or full stop, 35 thrips. And then we waited 3 days and then counted thrips every 3 days. Starting at day 15, we did count larvae, but I'm not going to quite show you that data. We'll just kind of talk about it at the end. But what this sort of looked like is there's a lot on this graph, but the idea is that we counted thrips every 3 days for 30 days, and that's on our x-axis. On the y-axis, that's the mean proportion of the 35 thrips that were recovered. So we'll take this one at a time because there's a lot of graphs here. But starting with the water treatment, and our treated plants are labeled in that red line, and our untreated plants were in the blue line. So for our water treated plants, if we put the thrips on the plant we treated with water, it basically stayed there from day 3 to day 6. But then by about day 9, you can see they're evenly spaced between an untreated and a treated plant. So there's lots of healthy leaf tissue in that area, so they're moving equally between water treated and an untreated plant. And they'll do that for the remainder of basically the first 15 days. Imazapyr, again, is a slow-acting herbicide. So on day 3, the thrips had stayed on that plant, on that imazapyr-treated plant. But by day 6, they were starting to move towards that untreated plant, and they remained there through day 15. So they were moving towards healthier leaf tissue. Not surprising with our Houdini active thrips, by day 3, they were gone from the triclopyr plant and they remained on the untreated plant for all 15 days. We found one surprising result, which was that our bark oil treated plants is that the thrips had kind of started to move away from the bark oil treated plants at day 3. And then by day 6, they were on untreated plants and seemed to stay there through the 15 days. Which was a little surprising, but when you look at what happened to it, is spraying the bark oil alone curled the tips of these plants. Just as a note, this would not be useful for a land manager at all because nobody's going to use bark oil on their own. It only serves as an adjuvant, and the whole goal is to kill the plant. So it's like, it's fun data, but it's not necessarily something that's like field useful because the whole goal is killing the plant, and that's contributing to it. So it just makes sense. Interesting stuff, maybe not useful. So from our kind of smaller scale experiments, we really saw that direct toxicity can actually overestimate mortality. They were surviving directly sprayed. We know that they can move away. We found our Pseudophilotrypsicini on these untreated plants, and not shown here, but we did find larvae on those untreated plants. So not only do they move away from the treated plants, but they will actually reproduce on the untreated plants. So they can find those healthy tissues. And I think this is a great example of a picture as we had treated all of these plants with triclopyr and all of that degraded leaf tissue, but the thrips managed to find the one single healthy leaf in the container. So we know they move. So because we know they move, it's a good reason for us to start that scale up process of looking at, it's a possible integrated management system for this system, so let's scale it up. And that's what we did next, as we scaled it up to the field study. And we did a few things here, but just to kind of review, we have these kinds of options for chemical control. And although imazapyr is the least harmful, the idea is that if we have to spray that over a tree that's 6-7 meters tall, we're not going to get an accurate herbicide coating of the foliage. So it kind of really left us only with one option to use triclopyr ester in the field. And that's what we ended up doing, was just applying the basal bark in the field. So the objective of the field study is determine the most effective approach for integrated management with Pseudophilthrip zucchini and triclopyr ester in the field. We have these two separate field plots. Just to give you an idea, 2 different locations. So in Okeechobee, Florida, this was in the back of a wonderful woman's ranch. So if I call it Miss Terry's plot, it's because this lovely lady generously donated kind of like the back of her land for us to just do all kinds of fun experiments. So that's Miss Terry's house in Okeechobee, Florida. And then the other one that we had was in Cypress Bend Preserve in Sebastian, Florida. And all of these kind of treatments were the same, is that anywhere there's kind of a white section, that was a plot for control. For the blue, we just did thrips by themselves. For the yellows, we did herbicide by itself, and only on female trees, which we'll talk about in just a second. And then for the purple one, that's a combination of thrips and herbicides in those plots. So when we set this up, we went into each of the plots when we scouted, and we looked for the male trees during the winter. So Brazilian pepper tree is dioecious, and only the females will produce berries. So the male trees would serve as excellent refugia for the thrips to feed on, because they're not producing berries and contributing to the spread of the plant, but they're also giving us this kind of nice source of food to keep our biocontrol agent alive, which is really helpful. So we used those. And at these plots, we did thrips releases at several different times. Basically, what we did was we just kind of tied a container using flagging tape to the trees to allow the thrips to move out of the container and do what they do. And then we did herbicide sprays in all the plots in December of 2021. So this is kind of how this went down, as we went out and we sprayed. So we did that basal bark application, and it took a little bit of time, but eventually it did defoliate those plants as we were looking for. And again, only the female trees is what we were aiming to defoliate. So we did a couple of different data collections, but the most important one, I think, to look at would really be the canopy coverage. So we estimated canopy coverage in each one of these plots, and estimates in increments of 10, but it's just estimating the amount of Brazilian pepper tree that was alive that was covering the plot. So we wouldn't include a dead one, and we wouldn't include something like the, palms back in there. And we have data collection all the way through 12 months on this one. We did have some site variation. So at Miss Terry's house in Okeechobee, it's just this wall of Brazilian pepper tree. So every one of our plots had like 80–90% coverage of Brazilian pepper tree in the beginning. And in Sebastian and Cypress Bend, we really saw these kind of patchy Brazilian pepper trees. So it was only 20–30%. So we kind of analyzed those separately because those sites were so variable. And we did find some differences. So the canopy coverage for Miss Terry's and Okeechobee is that there was a significant effect due to treatment and time. Part of that was seasonality, and I'm not exactly sure what the treatment is. When we separated those out, we had lower canopy coverage significantly in herbicide and thrips on their own, but it seemed to be highest when we had herbicide and thrips together. I'm not entirely sure what that exactly means, but that's kind of where we're at. I guess I should explain just a little bit better this graph as well, but on our x-axis, these are all the 12 months. So it's the initial setup, and then at 1 month, 3 months, 6 months, and 12 months when we collected those data. And these kind of colors correspond with the different treatment groups. So the black line was the control group, yellow was the herbicides, blue were the thrips by themselves, and then our herbicide and thrips together with a kind of purple line. And this is our percent canopy coverage estimate on the y-axis. So again, it's kind of all over the map. And so although we can get some results out of it, we're maybe not quite there yet. And I have some thoughts on that in just a second. When we did canopy coverage in Sebastian, again, the same x-axis with these kind of data collection points over the course of the year, and the canopy coverage here again, it's about 30%, so it's much lower in canopy coverage, but we definitely did not see a significant difference due to treatment or time here. It's just spaghetti noodles across the whole graph, so it's not telling us a whole lot there either. But I don't think that's the end of the story. There's some important parts to this. It's been discussed in both of the presentations preceding me, is that ecological integration is definitely key here, is that we got to make sure that we do something that keeps foliage available for our biocontrol agents to be able to feed, to be able to do what they do best, of reproduce and feed and control on their own. So making sure that they have a solid source of food is really, really key. Direct toxicities are really important to kind of get the setup going about what's going to happen. I would not have known or predicted as much the movement of the thrips had I not seen what happened in the Petri dishes. So starting small scale can be a really good place to kind of get an idea about what's going to happen later. For us, it also demonstrated the fact that refugia was going to be important, especially as we did those immigration studies. Longer data collection are often needed for these. Even in a year, it was hard to kind of tease apart where different effects were happening. But part of that is, I think, because we may not have had a lot of thrips out there. It's going to be dependent on whether it's a, more chompy feeder, like some of, trying to think, some of the beetles that are really taking the chunks out of the leaves, or seed feeders, whatever. Everything's going to be a little bit different. For thrips, they're tiny and they're causing a tiny bit of damage, and that's difficult for us to track. So we needed a lot more thrips out there. We started at 4,000 in each of the plots, and we kind of learned as we worked through some other studies that maybe the minimum we needed to release per plot was about 10,000. So some things needed to be adjusted there as we kind of figure out damage and biology and how these things are integrating out in the field. And then, as wonderful as this discussion is to have today, which is incredible, is that kind of the IPM stuff is going to be essential for controlling invasive plants. With globalization, there's no way that we're going to decrease the amount of invasive plants that we have. So our best weapon is to actually communicate with other resources. I'm a trained entomologist, and I didn't know anything about herbicides when I started this project, but I learned so much. And it is, some of the literature for herbicides is very different than what we do in entomology. And so trying to kind of weed through that and figure out how to integrate those two is difficult, and how to communicate with land managers who do things very differently than we do in a lab or in our field studies is really interesting ways to communicate. But getting those collaborations is really going to be key to make sure that we can kind of manage invasive weeds and their impacts a lot better. And with that, there's a ton of people that help to work on all of these projects. That's literally everybody. And I was with Dr. Mentier at the Indian River Research and Education Center in Florida. And all of this stuff was generously funded by the Florida Fish and Wildlife Conservation Commission down in Florida, which is amazing. But I'm currently at the University of Georgia as the undergraduate coordinator. So if you need to contact me, I do love to talk about this stuff. I'm with you guys on it. So if you need anything or have any questions, please reach out to me. So it'll be a much different conversation than talking to undergrads all day, which I also love, but it's a nice break.
That's great. Thank you, Kelly. It looks like we do have a question in the chat from Jane Mann.
Okay. Let me see if I can figure out how to, oh, hold on.
That's fine too.
Do you want to just read it? Figure it out.
Oh yeah, I can read it to you. It says, thank you, Kelly. In your field study, you treated only female plants with herbicide. Is this common practice for managers dealing with Brazilian pepper tree that is targeting females, or do they treat indiscriminately?
Oh, they absolutely treat indiscriminately. And so this was just a way for us to think about, is there a way that we can propose it to land managers as we could spray less and still leave these available? So it's more of a thing to try to propose if we can see it long-term working and we can really show data that makes sense, it's something to propose as a possibility. But no, absolutely, land managers right now is just, if it's a Brazilian pepper tree and they have chemicals, the tree is gone. So yeah, no, it's not a common practice, and that's a great question, but that's where kind of getting to know each other and talking about those things to offer those possibilities is really important.
Great, thank you. Are there any other questions? All right, next up is Jeff Mosley. He's with Montana State University. He'll be presenting on the integration of biocontrol and grazing.
While Melissa's bringing slides up, you have a copy of most everything there in front of you for those of you who are here. One of the first questions, pretty easy to answer, is are biocontrol agents in Montana compatible with livestock raising? And the answer to that's pretty straightforward. Livestock raising is the most ubiquitous land use. Almost every single acre of rangeland or wildland, grazable woodland forest, grazable forest is grazed by livestock in Montana, a very high percentage. And we also know that we have lots and lots of very successful introductions of biocontrol agents. So the question of whether livestock grazing in the management sense is compatible with thriving biocontrol agent populations, the answer is yes. If you start looking at targeted grazing though, where you're specifically trying to use livestock as a tool to suppress the weeds, then the question is a little bit less certain. You know, one of the reasons that livestock grazing in general is compatible with control agents is that the livestock do not eradicate the weed. So that host plant's always there, and target plant's always there for the host for the control agents. But when you get into targeted livestock grazing, where you're trying to specifically apply some pressure to that targeted plant, then the question's a little bit less certain in that case. And so we have been doing a bunch of work, and colleagues and friends before me had been working a lot in Montana over the years, integrating or using targeted livestock grazing first on leafy spurge, and integrating that then with a little bit of use, not a lot of research in this, but with some of the biocontrol agents for leafy spurge. And then we picked up some work and carried it on looking at spotted knapweed. And we spent about almost 10 years working out the grazing prescription to use cattle and sheep, cattle or sheep, and then in a couple cases actually integrating the two of those together to suppress spotted knapweed. And we got done with a lot of that work, and the question was, well, yeah, but we spent a lot of time, money, and effort and care trying to introduce these biocontrol agents. And we like your stuff here using livestock, but we're not really sure if we want to put these livestock in where we've already put the biocontrol agents. So we checked out the literature, and it really wasn't very clear. And based on Carrie's work, it's still not real clear. But so this is one of the few studies. In fact, Carrie even had this one cited in there, which is nice to see. But we took on a 5-year project looking at sheep and biocontrol agents on spotted knapweed. And this project was done not too far outside of Polson on the Salish Kootenai. So there's the citation if you need it or want it, and a whole bunch more details in there. But I'm going to kind of use this as a way to go through and just highlight some points. So, you know, if you step back and think, just as others have said before, there is this potential for additive effects, for this synergy, perhaps. You know, this cumulative stress hypothesis that if you hit the plant from different ways, that you add stress to the plant and it should work. And so, you know, above ground defoliation by livestock could just be another form of herbivory, right? Root damage inflicted by insects and seed head feeding insects hitting on the seed heads. So that livestock are just potentially another way to add to the mix. And these, you know, as you all know a whole lot better than me, but these have been very successful in spotted knapweed in Montana, especially Cyclopeonis and Larinus, maybe less so Agapita. But these are pretty much ubiquitous in any good stand of spotted knapweed in Montana anymore. So if you're going to introduce targeted livestock grazing, in this study's case, sheep, you're going to do it into a situation that most likely has pretty good populations. And so this concept, you know, of different agents, I think Natalie brought this up, that, you know, you can add to stress. And so all we're talking about doing is adding the same concept, is just taking a mammalian herbivore, right, and adding it to the mix. And would that help? Would it work? Would it hurt the insects or not? So 3 different treatments, okay? biocontrol insects alone, biocontrol insects plus sheep grazing in July, which was in the late bud, early flowering stage of spotted knapweed, or biocontrol insects plus sheep August, which is in the full flowering stage. Okay, so 3 different treatments with and without sheep. And then looking at the effects, and we'll just look at this real briefly, looking at spotted knapweed and looking at various kind of categories of it in terms of density. Okay, so the first one is plants. So total plants, total spotted knapweed density. And these results are after 4 consecutive years. Okay, so basically the sheep were there for about a week, 4 years in a row. Okay, either in July or in August. And the key things to look here is in terms of total spotted knapweed plant density, where biocontrol insects were working alone, we were down to about 104 plants, total plants of spotted knapweed per meter squared. We were down to 15 or 40 if it was grazed in August or July, in addition to the biocontrol agents. So dramatic difference, 86% fewer spotted knapweed plants in July grazed areas than with biocontrol alone. So in this case, you got the three triple whammy with biocontrol insects. The quadruple whammy really made a difference. Doesn't mean this wasn't making a difference, but when you added in the extra mammalian herbivore, then it became quite striking. In terms of seeds, again, this was mentioned earlier too. You need to find, if you're going to attack reproduction, you need to figure out how a given target plant reproduces. Well, in spotted knapweed's case, it's all by seeds. So if you can reduce seed production, you can definitely have an effect over time. All right, so here we are again after 4 years. Okay, 140 seeds per meter squared of spotted knapweed compared to 36 and 4. Okay, this tries to highlight down here that the biocontrol alone was doing a bang-up job. There's some other research out there that shows a thriving spotted knapweed population puts out about 9,500 seeds per meter squared. Okay, the biocontrol alone had taken it down to 140. That's amazing. Okay, however, other research shows that it takes about 160 seeds per meter squared for a spotted knapweed population to sustain itself. So even though taking this down to 140, right, we just barely got it to the point where it might actually be making a difference. This was mentioned before too, that suppression or control is not necessarily the same as meeting your goals, right? But here the differences are far greater. And so by this quadruple whammy, if you will, by adding a mammalian herbivore, we can take these numbers of seeds dramatically below the threshold needed to sustain. And this is where there's a big difference, and probably one of the reasons why adding the sheep made such a big impact is looking at the density of seedlings and juvenile plants. These biocontrol insects, of course, they mostly attack the adult plants, right? And they weaken them, makes them more susceptible to other pathogens, makes them more susceptible to competition, etc. And they attack and reduce, suppress seed production. Okay, but oftentimes any seeds that remain then reproduce, and those seedlings and juvenile plants don't get suppressed for a while until they get bigger enough to attract the biocontrol agents. The neat thing about sheep, particularly, less so with cattle, but some with cattle as well, is that these animals eat the juvenile plants. That, combined with the cumulative stress from above ground and below ground herbivory, that's what really makes the difference. 93% fewer seedlings and juvenile plants in July grazed areas than with biocontrol alone. So some of you may know Jim's story. hope you do, or at least you know of his work. And Jim helped us on this project. He has an area down in the Bitterroot Valley where biocontrol alone finally wiped out, essentially, the spotted knapweed population, and it took 30 years. And what we've been able to do here is do the same thing, but do it in 6 years by adding sheep. So a couple reasons why this works. And if you were, we've developed prescriptions, grazing prescriptions for spotted knapweed and leafy spurge, cheatgrass, doing a bunch of work now with wildlife habitat as well. So we have those if you need them. Well, a couple of things that, why did this work? Well, the timing of the grazing, just like the timing of the herbicide, the timing of the grazing makes a difference. We graze before spotted knapweed produces viable seeds. Okay. We do it late in the year. 20, 30 years ago, the idea was to graze early in the year when the knapweed was green and lush and growing. And we actually flipped that on its head over time with some experiments to show that the perfect time, the better time to eat it, to apply the grazing, is when grasses and forbs, desirable forbs, are largely dormant. At that time, right about late bud, early flowering, okay? Grasses are dormant, the desirables, forbs are dormant, spotted knapweed's still green and attractive and palatable. Also, at that time of year, when they're being grazed, then soil moisture is pretty much being depleted, so the spotted knapweed doesn't come back very fast. Sometimes it'll reflower, but we did some studies on that and showed that even though it reflowers at that time of year, the seeds never fill because it's too late in the year. And then we try to graze it before most of the seed heads have actually developed. And so the labrinus are not in the inflorescence. They're not in the seed head yet. In terms of the other agents, the root eating agents, most times they're below ground. But if they do come above ground, there's potential for incidental ingestion by the sheep or the cattle. But it's been shown pretty well that both Agapita and Cyphocleonis are pretty amazing animals, critters, and that when they sense disturbance, they like to be in the ground. They drop to the ground and they play dead. And so it's very unusual for them to actually be ingested. And the labrinus, if they are up on the seed head, when you disturb them, you probably maybe run into this when you've been collecting them, but they tend to disperse. And so they avoid being eaten, essentially. The other reasons then why this works, as I mentioned, the big part of is compensatory recruitment. So you've seen a lot of different studies done with biocontrol, and this is one of the challenges, is that oftentimes you open up the niche, you get better, you get some seed production, you get better seedling establishment, you get more vigorous growth. And so by having this quadruple whammy, these are all attacking the adult plants. The sheep are also attacking seed production in the adult plants. But here is the kicker, that they do consume those seedlings and juvenile plants. And so when you put all that together, this is why it works. And it has to do with the demographics of the spotted knapweed population. So, as has been mentioned already, each different species, target plant, has its own demography. But with spotted knapweed, this is why it works, okay? Most of the plants in the thriving spotted knapweed population are young, 2 or 3 years old. A very small percentage, are more than 6 years old. So, if you significantly reduce adult plant density somehow, whether it be herbicides, biocontrol, targeted livestock grazing, or some combination thereof, and you did that for 6 or more consecutive years, and you also kept the recruitment from replacing the adult plants that die. These plants don't live forever. Eventually, they're dead. So the population crashes. But it has to be done 6 at least 6 consecutive years, right? But if you do that, it works. You time it so it's compatible with the biocontrol agents. You time it so that it's compatible with the desirable grasses and forbs. Right? You time it to concentrate the livestock grazing intensity on the target plant. And so wherever you have lots of seedlings that tend to establish and transition to adults, then that's probably a good place to apply targeted livestock grazing in addition to the biocontrol plants. If you have areas where recruitment's really slow, then probably over time, the biocontrol alone will probably meet your goals, most likely. But we have a lot of places where that's not the case. And 6 straight years of targeted livestock grazing can make a huge impact, and then you can pick them up and move into the next spot. And I'll just wrap up with saying that, so we did this for 4 consecutive years. But we have one shining example of where this has been applied on a field scale. Some of you are familiar with Mannix Brothers Ranch outside Helmville and Blackfoot Challenge. And they've been doing this. And after 6 years, like clockwork, data shows the population crashed. And it's consistently remained, it's almost zero. The biocontrol agents are still there. There's enough for fusion, but we can't even find spotted knapweed in our plots anymore. And so it can be done. We've done it with spotted knapweed. There's not a lot of research, as Carrie pointed out, with other species, but following those same principles, I'm pretty optimistic that we could probably supercharge the work of the biocontrol agents with a little bit of integration, better integration with livestock. So that's kind of what I wanted to go through. Any questions that might have?
Thanks, Jeff. There is a question online first, so we'll get to these. Brenda is asking Dr. Mosley, how many sheep did you use? How big was the project area? And how many biocontrol agents? I lost connection a couple of times, so I apologize if you covered this.
Okay. Now, so in this particular study, it's a small plot study. There are 7 sheep in each of the little replicates to graze with. On the management scale size at Mannix Brothers, that's a band of about 1,000 sheep that go in and they move across the landscape about 3 days or so in one area, one pasture before moving on to the next. Let's see, what was number of sheep? What else?
How big was the project
Oh,
area?
so that particular study, that's small plot stuff. 4 reps, each rep I think is about, I mean, they were small, they're less than 8.
Okay. Great, thank you. And then questions in the room?
Yeah, I said 2 questions. One, I was curious if the data from the Mannix operation, is that published and available?
Not yet.
Okay. Is it going?
Hope to be, right? Yeah. And this summer, Mannix Brothers Ranch is hosting, they won the Environmental Stewardship Award for Montana, and there's a tour there in June, and maybe that'll prompt us to write it up. But it's a pretty neat story in that we did some of our, when we were working out the prescription for spotted knapweed, we did that on Mannix's. And then they became excited about it, and then they introduced it on a management scale. And the ranch then worked with a BLM employee and a couple of county agents to keep the management monitoring going all those years. And it's pretty neat that how about that? It actually shows what the research tells you it should. And so we need to get that story out there. So maybe we'll be prompted to get that done before June.
I was just gonna ask if this tour is like open for the public to go to, and if this project's gonna be featured, because there might be some people in this room or online that would be interested in attending.
I don't know for sure.
Okay.
Unless it used to be that it was invite only kind of a tour.
Okay.
I think that it's been expanded a little bit.
Well, if you hear and it is, let me know and I
Sure.
could pass information on.
But I do know that, if there was interest from the biocontrol community in Montana, I'm sure that, that a tour could be arranged and could see it separate, which might even be more fun. But
Any questions back?
So, do they just primarily target the knapweed or do they, what's left, what are the desirable forages?
so, the question was do the sheep, just target the knapweed? And no, not only. One of the things we do, we've kind of settled on, we did some studies trying to combine it with cattle because the folks with cattle really didn't want the sheep eating the cow grass, if you will, right? And so we tried different combinations of timing and who went first, et cetera. And basically what we settled in on is that the cows, if you're gonna have cattle too, the cattle go in first. The cattle they don't eat much knapweed, they eat surprising amounts. But they pretty much focus in on the grass. That makes the grass less attractive to the sheep. And then the sheep come in and they definitely do focus and select for the knapweed. But they eat some grass too. But the idea is that they took them grass when it's dormant and after the cattle have already gotten what they wanted. One of the things we tried early on was we tried putting cattle in first early in the season. We thought, well, we'll wait about 3 weeks for it to recover and then we'll put the sheep in. That was a disaster. Because the cattle took grass, stimulated regrowth. They came up, sheep went in, and the green grass was much more attractive and palatable to them than knapweed, and they just pummeled the grass again. So by having it later in the year and having the sheep come in either with cattle or right soon afterwards, that's what works really well.
Was there, and then is there a rest period after the cattle and the sheep or?
Yeah,
Sheep?
these are relatively brief grazing periods, a week, 2 weeks at a time, maybe with cattle, a day or two with the sheep, and then the rest of the year, for recovery. The one thing you have to keep in mind though, in this case, is that if you were to skip some years thinking to try to benefit grass recovery, you would produce a lot of seeds of knapweed in the meantime. So it seems to work best if you graze it, and we graze it lightly, but you have to do it for 6 consecutive years. If you skip a year or two, then you set yourself back. But another concluding comment on that is when we started out doing these, we focused in on wanting the sheep or the cattle to eat the knapweed, right? And we based our moves on waiting until they ate the knapweed. No, it didn't take us long to figure out. I mean, we're not smartest eggs in the basket, so it took us a while, but we were shooting ourselves in the foot. It was much smarter to focus in on the amount of grazing was happening to the desirables. So we base all the moves now on the amount of grazing that's on the desirables. Make sure we don't exceed the minimum threshold, even if they haven't eaten very much knapweed, right? So focused on what you want to keep rather than focused on what you're trying to get rid of. And over time, that has served us very well.
So when they used a band of sheep or whatever, did they keep them in a confined area short period of time and then move them out?
So the question was, how are the sheep handled on these bigger, larger areas? And they're herded, but they're not fenced. So they're herded, and so the density is fairly high. And this is about 1,000 sheep band that comes through, and so, but it's not. They don't denude it. They're not fenced in there until they take it to the dirt. Not by any means.
And the results were favorable, just like your study.
Yeah.
All right, there's one more question in the chat. We're gonna do that one and then if we could table some of these for later, we're cutting into our next slide. But the last one in the chat is, are there any plans to replicate this on a larger scale?
Well, we have many large scale projects with knapweed and leafy spurge ongoing in Montana that have just management level monitoring of the effects of targeted livestock grazing. I don't know of any others that have included the biocontrol monitoring as part of that, which might be something interesting to think about, because there are many management level, large numbers of sheep and cattle on large landscapes that are applying these prescriptions. Could show you some pictures and pretty dramatic, and it holds true for leafy spurge too. It's about a 6-year. You start really seeing the impacts at about 3 years, and by 6 years, the yellow is pretty much gone. And so they're, just the demography of some of these plants is not that different. But I don't know of any ongoing management level monitoring for biocontrol where the target grazing is occurring, and we're not doing any of it right now.
Okay. I encourage anyone and everyone, if you have additional questions, try and connect with Jeff either later today or over email. If you don't have that option, feel free to send questions to Melissa and she can help get some of those answers. Thank you, Jeff. That was awesome. Next up is Jane Mangold with Montana State University, and she'll be presenting on the integration of biocontrol with revegetation.
Good afternoon, everybody. Can you hear me okay?
We can see your slides too.
Great. Okay. So I'm going to talk about integrating biocontrol and revegetation, and I'm going to state a disclaimer right off the bat and say that I've never actually done any research in a very tightly controlled situation where we've been able to isolate the effects of biocontrol and revegetation separately and together. But I am going to share some examples of where that type of work has been done, and share some of some other research experiences I've had that have caused me to scratch my head about this question. So I'm going to start though by just kind of giving an overview of integrated weed management, and what we hope will happen. And this echoes some of what Carrie was sharing with us and Natalie to start us off this afternoon. But if we have a weedy parcel of land, we commonly treat it with whatever sort of tool of choice for dealing with the weed. We open up these available niches. And ideally what happens is the desirable remnant vegetation that's still there fills in those open spaces, and we end up with a situation, this example here, it would be native rangeland in Montana. That's we get a gold star and we're all happy with how our treatment has worked out. Where revegetation fits in to integrated weed management is if we're in a situation where we've created these open niches, but instead of our desirable species reestablishing, either the same weed comes back or a different weed or suite of weeds come back. And with revegetation, we're actively trying to fill those open spaces with species that we want to have growing there. And it's typically done with seeding. So when we think about this in the context of biocontrol, this is one of the things I've thought about as I was preparing this presentation is weed control with biocontrol is generally slower compared to some of our other methods that are more of an acute impact on the plant community. For example, spraying a herbicide or even hand pulling. And, I've often wondered to myself, like, I think one of the beauties of biocontrol is it does give the plant community more time to respond. So if you think about competitive relationships between a weedy invasive plant and the desired plants that may still be there, as that biocontrol works on the invasive plant, its competitive ability is decreased, and hopefully that releases desirable species that would still be there. So the positive is that desirable species would respond and have more time to do that compared to an acute weed treatment. At the same time, it's possible that secondary invaders, secondary species could respond in a situation where biocontrol has been the main tool of choice for dealing with an invasive plant. So putting this presentation together, I kind of went into the literature and was reviewing some of the literature that's out there, and I came across a few different studies where desirable species do come back in response to biological control. Just those papers and the target species and location of where that work was done. And alternatively, situations where the desirable species did not come back in response to a biocontrol. Again, showing example species and locations for that work. What I do want to say though is that this situation is not unique to biocontrol. We're not signaling out or biocontrol. We see this with other weed control tactics as well. So a couple experiences that I've seen in some of my revegetation research over the years in Montana. I just want to share a couple of those with you to kind of give you food for thought, because it's been food for thought for me over the years. And one concerns spotted knapweed. And this was a situation in kind of the middle part of the Bitterroot Valley near Corvallis, Hamilton, where years ago there was work, a bunch of integrated weed management work done where spotted knapweed was the target species. And in this research, there were multiple studies at this site, and revegetation was integrated with several different types of spotted knapweed control, including biocontrol. And we went back to these sites, 15 years after a lot of that work was done, and this was what we saw on the ground. We could see these strips. These are drill seed strips where different native and non-native grasses had been seeded. They're there 15 years later. But if you look just outside these drill seeded areas, the plant community was still dominated by spotted knapweed. Now, what has always made me scratch my head about this situation is, biocontrol is ubiquitous at this site, multiple agents included. And I've often wondered if the presence of the biocontrol for that long period of time isn't one of the reasons why these seeded species have done so well over time. So this would be a situation where the seeding worked, desirable species dominating those areas where seeding occurred. I've also been involved with some research with leafy spurge looking at long-term outcomes of integrating revegetation with leafy spurge control tactics. And we saw a situation where the leafy spurge declined over time, even in areas where we didn't seed, but the plant community in these areas is now co-dominated by non-native secondary, what we might call secondary invaders. And at this site, leafy spurge insects are also present, but it's a different outcome thinking about the insects and the seeding, with secondary invaders being present there instead of in those areas where those seeded grasses didn't take. So just a couple examples from my own experience that have really made me think, dang, I wish we could better understand the role of biocontrol in these settings where we've integrated revegetation. But it is quite challenging to implement and to find examples where biocontrol and revegetation have been implemented in a way that we can really isolate and control for the effects of each control tactic individually and in combination. And, bugs don't really follow our research protocols, right? We put them out there, we may try to contain them, but over time it's difficult to keep them contained and really try to isolate the effect of the biocontrol and then the biocontrol plus any other treatments that we do. So I think this is one of the reasons why we don't find more examples of biocontrol and revegetation, or even, biocontrol and herbicides, some of these other treatments that we've talked about already today. So in preparation for this presentation, I did a real quick literature review where I just combined the terms biological control and revegetation. And I just use the Agricola database. So I did this in about an afternoon. So it was a very uh, just skimming the surface compared to what Carrie probably took months for Carrie to do with the 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 work that she shared with us earlier this afternoon. But this particular search only provided uh, 22 records that were biological control and revegetation combined. So what I thought I would do is just highlight, I have 3 examples of these papers that were present out of these 22 records to just kind of give us some ideas of situations where researchers have really been able to, in a very controlled setting, combine insects with revegetation, and in one of these examples, herbicide as well. So we'll just go through these real quick. What I'm going to do is give you the citation, the target weed, where it was done, and just very brief overview of the details, and then share a couple result slides for each of these studies. So the first one was actually a study where they integrated insects, herbicide, and revegetation. And this was done with leafy spurge at 2 different sites in North Dakota. And this was a fairly long-term study, 4 to 5 year study period. And one of the reasons the study took so long, and I really admire the researchers for doing this, is that they started out by simply releasing the Aphthona beetles into these at these study sites. So the first year they released the insects, and this is pretty interesting. They actually applied an insecticide each year to non-insect plots to try to really isolate the effect of the insect compared to having no insects at all. Then about a little over a year later, they applied the herbicide imazapic to these plots. And then about another year after that, they actually seeded grasses into these plots. So they did this sequential, incremental application and integration of tools. And here I'm going to show you one of their data slides. We're looking at leafy spurge density. This is the first year. So all we have present here is the non-treated control in blue, and then areas where they released the insect, or plots where the insect was present. So that was year 2, 2 years, one year after release of the insects. No significant difference between treated and untreated. Year 3, we add the herbicide by itself in green, and then we have the insects plus the herbicide in purple. So a decline in leafy spurge density, no difference between either tool by itself. Year 4 and year 5 was really where they started to see interactions across the 3 different types of control. And now the slide gets a little more complicated. We're going to just kind of focus on year 5 here for ease of interpretation. The blue is the control. This is insects only, herbicide only, insects and herbicide, revegetation. We got revegetation in there now. And then this is insects and revegetation, herbicide and then revegetation, and then all 3 of them combined. And it was only in places, I shouldn't say only, but the insects and herbicide, or each tool combined with revegetation, or all tools together, is where by 5 years afterwards, they were seeing the most significant decrease in leafy spurge density. So, um, you know, uh, applause for the value of integration, including revegetation. The second example I'll share is a paper from Cutting and Hugh Goldstein, where they worked with the species mile a minute weed. And this research was done at one site in Pennsylvania, and it was a 3-year study. Now, I am not familiar with this plant. I know what it is, but I don't know a lot about its biology, nor about how it would typically be treated. The biocontrol that they used was the mile a minute weevil. And this was another situation where they used an insecticide to have non-insect plots present in their experimental design. And they applied that 2 to 3 times per year to keep the insects out of some of those plots. On top of that, they seeded a mix of 5 different native species. And in contrast to the study I shared with you just previously, in this situation, they actually applied the insects and the revegetation at the same time. So what did they find? Was there a benefit of integrating insects and revegetation? Here is one of the figures from that study. We're looking at mile a minute percent cover. So, going from 0 to 80%. And they looked at this 1, 2, and 3 years after application of the treatments. And they also looked at it within each year. They were collecting data in May and June, and then July, August, and September. So I know there's a lot of dots and a lot of lines here, but the open triangle is insecticide and no seed. So you could think about this line here as that line, if you can just make it out right there, as like a non-treated control. There were the biocontrol weren't there because they applied an insecticide and they didn't do any seeding. In contrast, the closed circles would be the combination of biocontrol and seeding. So the first year, the only treatment that reduced mile a minute below that of the control was the integration of the weevils and the seeding. The second year, that's still doing the best, but now weevils and no seed is similar as well. And then this would be like the control up here again with the open triangles. By 2011, all treatments performing similarly. There's still a trend of the combination doing the best, but statistically they were all similar to each other in terms of the cover of the target weed. Looking at what's interesting is that previous study I showed you with leafy spurge, they didn't present any data on the abundance of their seeded species. This paper did, which I thought was good. Here we're looking at the number of plant species, and again, they collected data in May and July for 2 years, and they split their seeded species into, or they split the species they were finding in the plots into introduced and native. So just to kind of simplify us thinking about this study, these bars over here would be the control. So no seeding and an insecticide was applied. On the far right would be the combo of insects and revegetation, where the weevils were present and seeding. So looking at just May of 2010, the only place where, well, I shouldn't say the only place, but really what we're seeing in this, the effect of the treatments on the number of plant species other than mile a minute, there was just a seeding effect. If we look at the following year, now we're looking May of 2011 and July of 2011, we do start to see a an effect of the biocontrol and the seeding, especially in July. You see the highest abundance of native species compared to not doing anything over here where seeding is combined with the insects. Okay, the final example I thought would be interesting to share with you is a paper that was published in 2008 by Ferrero Serrano and others out of University of Wyoming. And this was done with Canada thistle, and it was actually a greenhouse study, so quite a bit easier to control conditions. They didn't have to use an insecticide. They did use some caging, but what they did was they harvested plants from the field, and they used the stem mining weevil, Pseudorhynchus latura, and then they grew Canada thistle with needle and thread grass, which is a cool season grass, or alkali saccatone, which is a warm season grass. And so they grew them in competition and then added the stem mining weevil to some pots and didn't add the weevil to other pots. And here's a summary. This is, I think this is the only figure in this paper. But what we're looking at here is root biomass of Canada thistle, needle and thread grass, and alkali saccatone, and then the shoot biomass. And you should be able to notice right off the bat that the biggest effect was going on below ground. So if we look at just what's going on with Canada thistle, it's happening below ground. So this would be Canada thistle growing by itself, root biomass about 8 grams per pot. Here's Canada thistle plus needle and thread grass, about the same. Here's Canada thistle grown with needle and thread and the weevil. And this is where we see the biggest reduction in root biomass. And then here's just the weevil. Here's the effect of alkali saccatone, and here's saccatone plus the weevil, okay? So we saw the biggest effect with this cool season grass plus the weevil. And if you'll notice, looking at what did needle and thread grass do as a desired species, it was also what was going on below ground, no effect on above ground growth. It's all what was going on below ground. And same with alkali saccatone. And one of the quotes from this paper was that restoration effects can be complemented with biocontrol, although the impact will depend on particular weed species, grass competitors, and biocontrol agents involved. So this kind of led me to, I'm going to finish with this conceptual idea. When we think about integrating biocontrol and revegetation, kind of have 3 things to think about: the invasive plant, the biocontrol agent, and the seeded species. And I think within each of these 3 components, there's a lot to think about with each of these 3 components. And I've just tried to list some of them here. These, I'm sure you could come up with other ideas as well, but factors that may influence this. For the invasive plant, there's some there. The reproductive strategy and rooting structure, life form and life history, I think, are very important. For the biocontrol agent, where is it attacking? What is its site of attack on the invasive plant? What's its life history? How efficacious is it? Survivability, ease of dispersal and availability. And then if we're thinking about revegetation with a seeded species, there's a lot to think about as well, as demonstrated with that last paper out of the University of Wyoming, where the cool season grass was having a greater effect than the warm season grass, perhaps because Canada thistle is cool season and maybe there was more competition there. But I think this type of structure could really be used for helping us test hypotheses, and also for using ecology and all these different factors that might be overlaying each other to inform how we would apply insects and revegetation, or biocontrol and revegetation in the field. And with that, I am over time, so my apologies, and I will take questions, or maybe save questions for the discussion so we can keep moving along.
I'm gonna go ahead and say, let's save them, so we can get through the presentations. There is time built in at the end to have some discussion, so we can revisit some of this. So keep those in mind here in a little bit. Up next, we have Jeff Littlefield with Montana State University presenting on the landscape level case study of tansy ragwort in Northwest Montana.
Yeah, this study is my presentation. It's not necessarily grading different tools with biocontrol, but looking at more integrated strategies for weed management. We tend to approach weed management on more of a local scale, more site-specific management. And there's a number of different techniques and tools and strategies you can use. I like this one that Koch and Trumbo developed for musk thistle. Is probably back in the 1980s. And it kind of shows you a flow of different management options you can use for this particular weed. Biocontrol and chemical control is just really kind of a small component of it. But as we kind of ramp up our scale to more of a land management scale, we can use a lot of these tools, but in sort of a different manner. Carrie mentioned you'd probably see the slide again, and she was correct. I like this slide too. And as she was mentioning, often we get to this stage where things are in orange and red, and people realize, oh my God, we have a weed problem, and it's time for biocontrol. And we're under kind of a problem, not only with control costs being high, but in the area that's infested. So we're kind of behind the 8 ball. And sometimes I think that we're best off working with biocontrol at this lower level of the invasive scale. And this is actually one example I'm going to be talking about where the project began more down in this level rather than this level. Considering the time that it takes biocontrol to become established, go through the testing, you're looking at 20, 25 years or longer. So the earlier you can work on biocontrol, probably better off you are. Again, using an old example, tansy ragwort, not to be confused with common tansy. It's introduced back into eastern US and Canada back in the mid 1800s, made its way to the East Coast early 1900s, eventually started working its way eastward again, found in Montana back in about 1980. A lot of these infestations down here are probably misidentifications rather than actual plants. Looking at the potential area where tansy ragwort can be invasive, this is from the Montana Natural Heritage Program. And you can see where the main infestation of tansy is, pretty much up in the northwest corner. This kind of shows you a kind of a really nice patch. It can be a moderately aggressive weed. It's a biennial. So the first year it's meant as seedlings, fall rosettes, which overwinter. Next year, bulbs produce flowers, produce maybe a couple hundred seeds per plant. And those are dispersed either by wind or in hay or equipment or even animals. If you disturb the bolted plant, it becomes really kind of a short-lived perennial as well. One bad thing about it, it also has a bunch of alkaloids in it, which is toxic to particularly horses and cattle, not so much sheep and goats. And this causes liver damage over more of a chronic situation where it builds up over time. It has a lot of compounds in it. I kind of really like the English common name. I think it's from England, Stinking Willy. I always thought that was a good name for a weed. Problem in Montana has been mostly up in the northwest corner between Lincoln and Flathead County. Back in probably the mid 1990s, the Little Wolf fire came through on the Lincoln side, going over to the Flathead County side. And at that time, they found after the fire a fairly large infestation of Tansy ragwort. With additional survey, they found larger areas to the southwest, plus a lot of scattered infestations through an area. And basically, they came and did an outer perimeter. This kind of forms a core of the main Tansy ragwort area at that time. And there's probably about 500,000 acres in that group, comprised of a variety of landowners, Forest Service, ex-Plum Creek, private land, DNRC, quite a variety of landowners with different management strategies and different control resources, I would say, for managing it. So the main problem is you have a weed over a large area, multiple landowners. So they were trying to come up with an area-wide management plan to contain the present infestation and reduce the spread in seed production. So, and also to develop kind of a long-term strategy to reduce the core population to kind of an economic and ecological acceptable level. So some of the first things they did was to develop a weed management association. So they just delineated the size of the infestation, identified who the main players were and potential cooperators in this area, and basically formed a task force group. So the purpose of the task force was to obtain funding for control. At that time, Tansy ragwort was not a designated noxious weed. So they got that listed as a weed. And also they created a program coordinator, which is instrumental at doing a lot of coordination plus some of the treatments. So the early coordinator for a number of years was Bill Jalvin here. He's just retired a couple of years ago. So he's been with the project for a long time. And what does Bill do? He did all the work. So, but a lot of the coordination and survey work, a lot of emphasis, since it was a new weed, placed on education and awareness. So they had workshops, they had signs built as you go into the Tansy area to inform people. They developed some initial control strategies, suppression of the core infestation by aerial spraying, backpack spraying. They contained the perimeter of the infestation by closing off road systems, especially secondary roads, prevent mostly public from accessing the area and spreading the weeds outside the perimeter. They implemented biocontrol, did a lot of monitoring and mapping, which they're still doing today. Needless to say, not all the control tactics were favorably embraced. So, especially road closures. That was during hunting season. That was a bad thing to be up there. They also initiated a research program looking at herbicides, timing, determining the weed ecology, how it differs from other places, especially in the Pacific Northwest, Oregon, Washington. And they also tried to determine if the biocontrol agents that were available at the time were effective or could be used in Montana. Biocontrol has been one of the more successful programs in the Northwest, very successful in Oregon. Began in the 1960s. There's been 3 agents approved and established that were available for use in Montana at that time. The first agent that was brought in was cinnabar moth. The adults emerged kind of mid-summer, seek out fairly large patches of Tansy ragwort. The larvae can defoliate the plant, reducing flowering, seed production, and preventing seed set. Has one advantage in Montana versus Oregon is that in Oregon, there's a long enough growing season so the plants can recompensate and put out additional seeds. Where in Montana, the growing season's a lot shorter, so it has more impact on the seed resource. And eventually became fairly well established. One of the main concerns is that it's non-target feeding. In Oregon, it feeds on 2 native species, kind of limited feeding in Montana, but it's certainly a concern. And that's one of the reasons why you can't move it around from state to state anymore. It's been fairly effective at reducing plant populations, although there, like biocontrol, there are rebounds in plant populations. This shows you kind of the initial site over in the flathead side, before and after, for probably maybe a good 10-year period. You cannot find a tansy ragwort in this fairly large section of Flathead County. Pretty much eliminated tansy ragwort over a fairly large area. And it's only been recently where plants are starting to reinvade. The second agent that's been also successful is the flea beetle. This is the most successful biocontrol agent in Oregon and Washington. It attacks, it's very mobile and can attack really kind of small patches where cinnamons attack larger plants, patches. These can attack smaller patches of tansy ragwort. They attack mostly the rosettes in the fall time. In the following spring, they lay their eggs on the rosettes. They either overwinter on the rosette or overwinter as larvae. And they basically wind up in high populations, killing the rosette or weakening it, so it has stress the following year. We wound up releasing 3 different biotypes of population in Montana. 2 of them established. There was a high elevation population brought in from Oregon, which eventually established, but we also brought in a third biotype from Switzerland that's more cold adapted, and that was really established. But at some point in time, those 2 have hybridized. So we have hybrids now in certain areas, and those hybrids tend to be more vigorous than the parent population. It was really kind of slow to establish compared to cinnabar, but now it's fairly widespread. And it kind of goes through kind of a 3-year life cycle of boom and bust, depending on where you are. They have a tendency to burn slash piles up in that area, and it creates a really nice seed bed for mostly weeds and tansy ragwort. And in that situation, they build up these really high populations. We have maybe 800 beetles per single plant. Really high populations, where more of a field situation, the population is much lower. But kind of that boom and bust type situation. Gives the example, some before and after pictures. It can also kind of eradicate the weed in certain small patches. The third agent is seed head fly, which established well, but never really built up populations to impact seed production at all. So that's not really, it's fairly widespread, but not being utilized as biocontrol. So we have 2 agents that are fairly effective, and fortunately they are somewhat compatible. This is one of my field sites, a little wet area surrounded by tansy ragwort. We put out flea beetles, and as they started picking up populations, the cinnamons found this patch, and probably within a 2-year period, it looked like this. There's still a few small rosettes around that are being fed on by the flea beetle. So the combination of the 2 has been fairly effective as biocontrol agents, attacking different stages of the plant, different patch sizes. So with the program, some initial results. There was substantial control, both from chemical control and biological control, in the kind of core area that had fairly high tansy ragwort density. That allowed the redirecting of efforts to more peripheral areas, looking for outlying populations, treating plants along vector corridors, roadsides, and things of that sort. However, new infestations have been found in Montana, kind of over by the town of Lincoln, up in Glacier National Park. So why was this program was reasonably effective? One is that there was a well-coordinated strategy, and there was also a buy-in with the local cooperators. So more of kind of a social interaction of buy-in, even though they weren't 100% sure on some of these management strategies. There was early suppression of ragwort in the core areas and peripheral areas, and part of this was due by effective biocontrol early on in the program. Also, it's successful because tansy is sort of a moderate aggressive weed, can find certain more open habitats, more disturbed habitats, and it's also a fairly new invader, so it's not really super widespread across the state, kind of more localized in the northwest corner. And then also there's a commitment for long-term management, and more importantly, there's been continued long-term funding for the project, which has really helped. So with that, a lot of the initial cooperators associated with the project, a variety of people who worked on it in different aspects. And I think I have time for, I don't know, maybe a question or two.
Yeah, are there any questions for Jeff?
Now, did you say that two types of weed beetles hybrided together?
Or they're basically the same species. So basically these populations differ in how they overwinter. So the Italian populations tend to hatch out in the fall and they overwinter as larvae, whereas the Swiss overwinter as eggs. So at some point they have overlapped and have hybridized. So we have kind of a combination of the hybrids. Some of them hatch out in the fall, some of them overwinter as eggs. And they seem to lay more eggs, have a lot more offspring, a little more vigorous. Whether that will continue through time, it's not sure.
Are there any additional questions? All right,
Okay,
thanks,
thank you.
Jeff. Thanks to all of our presenters. I don't know if you want to cut into our group discussion time to address maybe some questions we asked.
Well, that would be fine.
Okay.
Yeah.
So we're gonna kind of readjust and give time to ask some of the questions that we skipped over to get for presentations. So the gentleman back here had a question for Jeff Wilson.
Oh, I was on that ranch, you said 1,000 sheep. What size of acres were they sitting on for 6 years? Because earlier you said something about 6 to 7 sheep on an acre when you guys were plotting and contesting in small area.
Yeah, so, boy, I don't know I could look it up, beauty. I don't know I can't remember what our recommended stocking rate is, but those on large scale projects typically would have about 1,000 head sheep. They move through an area and they might be I mean, the length of time they'll stay there depends on the amount of weed as well as the amount of the grass. They typically won't eat sheep won't eat more than about half their diet as an athlete. And so how long they stay depends on how much grass is left. But typically the sheep are moving on about a 3-day schedule. And so they might move through on that one particular ranch. A lot of the pastures are that's where you're from, reasonably small. So they might only be a quarter of the section. They might only be there a few hours. So they move through, and then you get to a large pasture, they might be in there for a couple days. But the reason it's successful is focusing in on not taking the grass down below 3 to 4 inches. and the fact that the grass is being grazed when it's dormant, and very tolerant of being grazed. And that set up the grass very well then to respond favorably to the reduced competition. Yeah, that's, I could give you some more numbers and things.
I had a question for you, Jeff, and I might be mixing this up with purge, but I've been told once that when you do targeted grazing, that you kind of have to train sheep or goats to feed on those particular weed species, that it doesn't, you can't just necessarily go buy a sheep and throw them on there and think they're gonna be affected, like it's kind of a training concept, is that accurate, or?
Yes and no. There is evidence that there's definite evidence, we know this, that there are physiological differences amongst livestock. And there are physiological differences in their ability to handle toxins. And so it stands to reason that there are some animals that are better predisposed because of their breeding to handle certain feeds. But you can't train that into, that's genetics. And so my colleague, former colleague, Brett Olson, who's retired now, he did some work years ago on that because there were some folks that were trying to market sheep, spurge eating sheep at a premium. And what they figured out is after about 3 weeks, the trained ones versus the untrained, they were eating the same amount. They picked it up. But this idea that there are some strains or some herds out there that maybe are better suited, that's true. And we did some work with Texas A&M and showed that's true on, they use goats to control juniper. And physiologically, there are some goats that don't let the toxins into their bloodstream so they can eat more of it. And that's a genetic thing that then Texas A&M has actually bred a herd of super juniper eating goats because of that. So there are some things that are true there, you know, and then this idea of training animals to eat them. We studied that with cattle and hapweed. I think it's one of the few studies anyways, it's actually studied it. And the answer is that cattle can be trained. You can follow that technique. Definitely will eat knapweed, but they don't eat any more than the ones you don't train. So I guess it works, but you don't need to do that work.
Thanks. Are there any additional questions online from any of the presentations? We may have lost some of our presenters too.
I think we did.
And if that's the case, and you have one for someone who's not with us, we can try and pass along your question. Any more questions here? Right, we're gonna move on to our group discussion, which is intended to be focused on addressing possible research needs, educational materials that we may be lacking as a biocontrol community that we could create or pass along, and the like, etc. Whatever you want that to be. Yeah. That's the time to talk about it. I personally really like the stickers, and I think we should have more stickers. That's my opinion.
So Hannah, you had mentioned something before in our conversations of kind of like a handout that you thought would be beneficial along these lines. Would you like to describe
I
that?
don't even know if I remember. Can you remind me?
It was,
Oh, wait,
it
I
was
think,
about, okay, yeah, you go ahead.
okay. About timing, is it related to the timing and?
Compatibility.
Yeah, all right. So some of us have been to MWCA conferences, and I think it was Bear had it was what, 2019, when Esplanade still had its special use application, but they had a diagram tank mixes and timing for pre-emergent and post-emergent. Does this ring a bell to anybody? It was almost like these little bar graphs showing that if you're gonna do a pre-emergent and post-emergent tank mix at this time, this is the recommendation in a given season. Does this ring a bell to anybody? I've not been able to find it online, I'm sorry, but I thought it was a cool concept because when we're talking about revegetation, grazing, which you kind of touched on, July and August differences, herbicide application mixed in with biocontrol, I was imagining a similar diagram that would show recommended timing for some of these integrated approaches. Like, it's best to integrate grazing with biocontrol during these windows, but maybe not this one, to give people better guidance on when they can implement certain types of integrated weed management approaches. I don't know. Yeah. That's just it. Too bad I don't have the diagram showing that.
I do like that idea, just as far as like public outreach component, because I think a lot of, I don't know, I get calls all the time from people that want, Nappy Weevils and it's October, or want Nappy Weevils and it's.
The poster kind of covers.
That, but like having some kind of Gantt chart for some of these species, like, this is when you should be collecting these insects for distributions, when you should be integrating grazing. It can be hard to, you also have to do it for each species.
Well, somewhere where those like release efforts on a biocontrol level are not compromised by the application of another tool. So something that's gonna support both working together.
Yeah, I think it would have to be species specific, you know, right? 'Cause
Yeah.
it's so different from one species
Yeah.
to the other. So like, you know, there's a lot of conversation about how potentially grazing and toad flax agents aren't compatible. Or if you are gonna use grazing and toad flax agents, you have to really push those animals, so they're just like going after the flowers and not munching down into the stems where the insects are. But that's very, and Jeff mentions that too, about the seed agents for spotted knapweed and grazing. Like how you have to do that timing. So it's not necessarily that it's not compatible, but you have to really think about timing for some of these things.
Jane, if, you guys can probably read that, but, her comment in question, there are a lot of noxious weeds and biocontrol agents. Would the handout cover the most common noxious weeds and biocontrol systems then, and those that we have research for? I guess in its most simplest form and idea, yes, it would be kind of the more common ones, especially in Montana, that we're focusing on. So like toad flaxes, knapweed, and spurge, kind of the big ones we collect for. I wish Teresa's here. I don't know if she's online. Teresa, are you online? She's here. She was at one point. She might have that handout.
Oh, she is there
Somewhere.
She just chatted.
Teresa, have you seen the handout that I'm talking about? And if you can find it, if you'd like to share it with us. Or if we could reach out to Steve Saunders, maybe he knows where it is.
You know, the other thing that strikes me is putting that together would also be a great way to highlight what we don't know, you know, maybe which species or which agents, and lead to setting up
Some gaps.
projects, identifying gaps.
Yeah. And I think that some of the discussion we've had about how people reporting post-release monitoring over time, like people are implementing biocontrol under all kinds of different management conditions across states. And so maybe highlighting already data that we're already collecting in areas, seeing if, you know, are there correlations that could be tested in the landscape already, because we are collecting these data as these things, as these insects are distributed.
Was there anything that jumped out to anybody, as like a gap in research that we would really like to see a project on integrating?
I don't know if I have like a specific project, but I'm kind of intrigued by this idea of providing refuge for biocontrol agents in these systems. And in Missoula Valley, we've definitely seen sites where the Dalmatian toadflax weevils have been really successful, and the manager has gone in with the intention to kind of like hopefully eradicate that. And clearly they were unsuccessful. And what they did instead was remove the last refuge for the insects that were in that site. And then had a huge resurgence over a number of years of Dalmatian toadflax and not have the biocontrol in check. So I'm really curious about this idea of trying to integrate that concept of refuge into management recommendations if we're really focusing on effectively integrating biocontrol over a long term. But I have no idea what that project looks like or how you effectively convey what refuge is necessary or I don't know what project is, but my brain was doing out there.
There's a social component to that.
Yeah.
Because I've been places, where the weed is pretty much controlled biologically, but there are still plants there. The philosophy is to get those last plants, because that's what they're paid to do. And the same thing, you eliminate those refugia. And, it's just changing the mindset of people. That probably impossible to kill everything. But there are cases where it's beneficial not to.
Yeah, I mean, maybe there's not a research project there, but within the recommendation documents, like value of that refuge and those managing a certain threshold of then eradication is probably marked. Yeah. You know.
A lot depends where those refuges are located as to whether they should be treated or not. Overall management standpoint. They're on, say, a vector of some sort.
Yeah.
I don't know.
I think that's a good point though, like talking, we talk about, what's the threshold at which we consider control is, what the method of control functions, right? And it's maybe talking about it as like flipping it around, we talk about those thresholds anyway, but that's as low as the management will get. Maybe flipping the conversation a little bit to say, that's where the beetle population can push it down, and that's where the beetle population can stay, and that'll create like adding that, so taking that conversation and tweaking it a little bit into this refugium kind of concept that speaks more to the perpetuity of biocontrol. Especially in terms of these cycles that have been, like people have talked about today, you have these waxes and wanes sometimes in the same areas. But if I got eradicated down or like down to the level, but over larger scales, that's what's happening. And you're having kind of.
Well, I think it goes back to, we talk about flipping things around, like what you're saying as talkers. Getting back to the monitoring and focusing on species are there.
I think it is a good point. That is somewhat of a social dilemma.
This information may be out there. This isn't my specialty by any means, but I can see where if it's out there, if you could give somebody some numbers, like this is about the density of plants. This is about the distance from, how far away this refuge could be. I mean, especially when you start talking about integrating tools like herbicides or hand pulling or whatever else, that's where you get into the very last plant. right? We're gonna use both tools, we're gonna let Biocontrol take it down, and then we're gonna get to a management point, and then we're gonna really hit it. And so if that's shooting yourself in the foot in the long run, that'd be good to know. You know, like, yeah, you can go out there and hand spray, spin it, but don't go below X amount. And I don't know, maybe those numbers are out there, but right now I don't see that, but
I mean, it's realistic, though, it's an actionable threshold of what you want to do. I mean, we do it the other way in our programs with grasshoppers, certain densities warrant intervention, and until that's met, I just leave it alone. Well, I think some of that's demographic. We're discussing some of more demographic, but a lot of times we don't have those. I know, like in the Team Leafy Spurge era, they've tried to put those kind of numbers on things. I don't know if you've seen those they've got those long handouts. That was one thing they tried to give you, like rules of thumb for like knowing when you have an established population, what could be considered control. They tried to put some kind of characters on it, but that still isn't precise. We have a couple different topics in the chat. Do we want to table this one and kind of think on it some more? Hopefully Jane can find that stewardship guide line. It might have that diagram in it that I was talking about. Looks like she's sharing it.
Yes, so this is the stewardship guidelines for Esplanade. I don't know what the date was, but there's this treatment recommendation for different growth stages. And then if you scroll down, there's kind of a calendar recommendation that's broken up by month of the year. Hannah, is this what you were thinking of, Hannah?
But then taking this concept and applying it to integrated approaches with revegetation, grazing, herbicide in conjunction with biocontrol. So, yeah, this is it. This is it. I'm so glad you found it. It's kind of going crazy on us.
I have it. If anyone, I mean, if there's a way, if I can send it to anybody, so that you have the PDF, let me know.
You can just drop it in the chat box.
Sure.
Can I drop? This is actually saved on my computer.
You can usually see in the chat. Click those three buttons, three dots.
Okay, I'll see if I can figure it out while we're talking here.
Able to drop things in the chat and Teams.
You can also email it to me, Jane, and I'm happy to share it with anybody who's interested. I do see that Brenda asked if you could share it with her.
How about I send it to Melissa and Melissa sends it to the group? Would that be best? Okay.
Oh.
We have now kind of shifting gears. We have some comments from David Weaver. Are there any current efforts to document the riparian distribution of waterway invasive Russian olive? And how severe is it? And this reasoning is integrated weed management of salt cedar may simply facilitate a more severe problem with Russian olive.
I'm looking at you, Joe, because I think you guys do quite a bit of this. Is there like a mapping effort for Russian olive when you guys are treating it?
We don't treat Russian olive.
Oh, you don't?
No, we don't.
Oh, it's salt cedar that you've been
Salt cedar, yes, and we have our salt cedar that we do treat.
There was that Montana, what maybe was the natural here, did the Yellowstone. But that was in the early 2000s. So I don't think there's any, I don't know of any current efforts.
Well, there's that pretty invasive.
Yeah.
Put together. I think that's one of their goals is to grab what everybody's got so far, see what the gaps are on all three with buckthorn and with all salt cedars.
Yeah.
I almost wondered if you could do it with satellite stuff.
Yeah.
Pretty charismatic.
Yeah, it's a pretty unique color. I don't know, maybe there's some willows that are large too, but
Have you noticed, Joe, with your treatments of salt cedar and influx of Russian olive as a successive species?
No, I have not. No. What I've been noticing is going back to following here and looking at some of those sites, we tend to see a little bit of Canada thistle come in where there's bare ground because what we're using sometimes creates a dead ring for about a year.
Is there Russian olive in the area that could move in?
Yes, but I haven't seen that.
Yeah, I thought Kelly's research was of interest, you know, particularly because of the woody invasives grant that was just funded and this idea of this statewide effort to treat a number of these species, and because, you know, David and Charlene have been doing a lot of work to develop these biocontrol agents for both Russian olive and salt cedar. So I think it's really important for us to think about that integration because, you know, I shouldn't speak because I haven't done any of the herbicide treatments, but my understanding is herbicide treatments for both salt cedar and Russian olive is pretty resource heavy, takes a lot of time. It's not really possible to do all of it.
Russian olive, I don't know where you're gonna find the money now to do that.
Yeah.
Salt cedar, we might as well, let's keep going while we can and.
Yeah.
Before that one gets to be a beast.
Mhm. Yeah.
I'm afraid it might be in some areas.
I think it is in some areas, yeah.
That's why we're gonna have to seek other funding. Cause I'm not gonna be able to go back to the trust fund. I can't go back to the NRC now, so.
Yeah, so that also speaks to like integrated tools.
Yeah.
So I think it's just really important for us to start thinking about those things, maybe not necessarily really ahead of time, but you know, kind of in the beginning.
And one thing I was thinking about with the various talks is that there's been several times that the point made that we need to understand the larger scale, like a larger scale outside of the plot scale. And that's one thing a group like this could facilitate is if there was project or questions such as this that this group could help facilitate like connections that would allow us to have a larger scale of information or monitoring effort for some sort of integrated question.
There's some additional comments from David and Charlene.
And there's somebody else.
As Brenda.
It was Brenda.
Brenda, I wanted to clarify, were you extending that offer to David to share those results?
Sorry, yeah, I'm more than happy to share what we did. It was to Dave and Charlene or to whoever. It was we were a multi-county BLM blah 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 task force in, I want to say, the mid to late 90s, and then no, mid to late 2000s. I'm sorry, not that old. And then the salt cedar and Russian olive project that we did in Treasure County was a multi-phase project that was started in 2004. And I have extensive data on that. So I'm more than happy to share that.
Awesome, thank you. Dave proposed question, could designating Russian olive as noxious in riparian habitats help allocate more resources? And I think the emphasis there, and you can correct me if I'm wrong, is strictly in riparian habitats, because I know there's some controversy around listing Russian olive entirely as a noxious weed in all habitats. Is that accurate, David? And are there any thoughts on that?
Yeah, that's accurate.
Yeah, that was my question. I don't know if Jasmine's still on, but I'm not aware of any species that we've designated by habitat type or certain geography. I don't know if
She
it's-
is.
Yeah, she's.
I'm still here, yeah.
Your thoughts?
Yeah, I don't actually know if that's possible. I don't know. I've never seen it. I haven't seen it in other states. I know we have never done it, but it's worth talking about, I guess.
Russian Olive is listed in those counties, is that what you're saying?
I know it's for sure listed in Treasure County, but I thought it was from Treasure County.
Yeah, that's the only county, and that's kind of the alternative is the counties where they have it in riparian areas can list it, but it's county-wide.
You could maybe call it the resolution that's.
Yeah, I mean, because of shelter belts, it'll never go.
Yeah.
You're already done.
Yeah.
But it's still being done.
It's still
I
getting
live in
sold.
East- Yeah, I live in Eastern Montana.
Oh, that's not good.
We don't sell it. We don't have to sell it.
In addition, Charlene, so that's too bad about the decades of effort and state funding invested in salt cedar biocontrol. Charlene, if you want to clarify a little bit or expand on that. Might help.
Sure, what I heard is, people are charging ahead with salt cedar management, regardless of where it occurs, as long as it's riparian, and, you know, that's where we have our biocontrol stuff set up, so we are just going to throw away all those years of research, and state funding in this program in order to spray stuff? Or maybe I'm just misunderstanding the comment.
Yeah, I don't know. I think certain areas have spray programs, but I don't think it's very extensive. Like, I don't know, Joe, maybe you know more. I know Yellowstone County does treatments for salt cedar, but I don't know. Are there a lot of other counties that are doing that?
No,
Or-
there's not.
Yeah, I didn't think so.
I think Treasure County played around with it for a little bit, but there's no sense in them really getting too involved in it until we gotta get our county cleaned up. And then Bighorn County need to start a project. Because with the Bighorn, dumping into the Yellowstone down there at, Yellowstone Treasure County line, they're gonna pollute them.
That project Brett was talking about was basically Yellowstone County.
Yeah, I think that data could be fairly revealing of, I mean, I think the underlying question is are any management efforts made on salt cedar going to make a more severe problem with Russian olive as a successive species? You have not observed Russian olive being a successive species, but maybe their data set has some figures that could suggest otherwise. I don't know. It looks like Brenda, you have your hand up. Do you want to?
Well, I guess just a couple things. So, Kenny is correct. Years ago, when the attention started getting put on Russian olive, Treasure County was the one and only county, especially in eastern Montana, that had political input and support to list Russian olive as a county or as an invasive species in that county. One thing I need to clean up or confirm, I can't think right now, it's late in the day, is that island that we treated in Treasure County that we did the Russian olive and then and the salt cedar projects on, that was a fuels reduction. It was driven by a fuels reduction project. And basically what had happened is it was an island, one of the very few islands that BLM actually has. And basically the Russian olive and the salt cedar had encroached so much that the meadows were no longer existed. And I have documentation and pictures and videos and stuff where basically the wildlife had to make holes through the Russian olive and the salt cedar trees to get back and forth from the island, the meadows, the small meadows that were still on the island out to the water. And so that's what spurred that. And then the Salt Cedar Task Force group that I was on, it wasn't Yellowstone. This was prior to that. This was the counties from the North Dakota line through my field office. So it would have went through Treasure County. So from Treasure County through Richland County, we had all of those counties involved with the BLM and the DNRC. And we had hired a contractors, and then we went county by county to treat the salt cedar above the high water mark. But like I said, this was years ago. And I think, Charlene, this was prior to any releases that were put on the river, because Kenny and I were involved in the tent making process in 98 at Lovell. And I don't believe, like, I know I put in a few years ago, I put in for some, like 6 different sites to be studied over here. And so this was long after that task force. So if that was your concern, they were in 2 different areas and 2 different projects. So I don't think that the treatment would have affected the sites, the study sites that we had put in for, if that makes sense.
Yeah, so Charlene, are you speaking more to a concern about the effort that's gone into biocontrol for salt cedar, not for Russian olive? I think is what I was understanding. And you know, again, I'm not an expert on salt cedar, but my understanding is that there's plenty of it, and it doesn't seem like it's going to be plausible to treat it all with herbicide or manually. There's always going to be areas where these biocontrol agents are going to be needed. I think it's just all about communication and making sure that, you know, the people who are doing herbicide treatments know where they should not be treating, so that, you know, we can continue to work to get these agents established.
Yeah, exactly. And better communication, i.e. me understanding what's going on, would help it. My apologies to everyone. I was conflating this project that is being discussed today with the 4-county effort, the woody invasives thing. And yeah, I'm just concerned about our biocontrol sites. That's all.
Yeah, I think it's really important that we engage in that effort, and you know, whoever the contractor is that they hire for it early, and make sure that when that plan is developed, that, you know, your and David's efforts are not undermined with other treatment methods. And Kelsey was on, I know she was a big part of writing that grant. I'm not sure if she's still on.
Yeah, I'm
yeah.
here. I've been listening in, and thanks so much for the feedback. I was going to message you, Brenda. But absolutely, I think you put it really well, Melissa. And that's kind of the hopeful function of the task force, is to assimilate all these people and all this information so that those efforts moving forward can be effective and efficient.
So of course, our dream is that the salt cedar agents will become well enough established and expand and be successful like they have been further south. They've been bottlenecking for years. And so I guess that would be something we wouldn't want to have happen, is if we suddenly see a large expansion in the populations, to think that we need to devote precious resources for managing riparian dominating wheat species into something that we have an alternative control. Because certainly we can apply chemical control to Russian olive, salt cedar, buckthorn, as outlined in the woody invasives proposal.
I think we have some interesting things to think about. We can keep talking unless we do have time on the agenda for other business. So if we know what other business is, I don't have any other business, we keep talking, but if you have other business and you'd like to talk about it, we can change the topic.
I do have one other thing that I wanted to bring up, but I think before I do that, I just want to make sure that David and Charlene feel okay with where we just left that conversation. You know, it sounds like there's a lot of interest in making sure that you guys are engaged with this new woody invasives task force. Is that a reasonable step forward, or is there something else that you feel like should occur in addition to that at this point?
So like everyone else, we don't like woody invasives at all in the riparian habitats. What I was trying to say is that if we have some potential to help the thing with better progression of salt cedar biocontrol, that may allow for more resources to go into the other species where we're not going to have biocontrol agents that are going to feed on them. But yeah, I'm happy with where the conversation is. I just wanted to put it out there because I found out recently you can't even find out how bad Russian olive invasions are, but everywhere we're doing salt cedar work, there's giant Russian olive trees that have been there for a long time. And it seems like we're just maybe looking past it a little, even though it's maybe getting some attention now in the Woody Invasives Task Force. So that's the only reason I brought it up.
I also just want to clarify something. I maybe sense some overlap. So the task force, as in the trust fund grant that was made for planning, doesn't have any money in it for treatments. So that's just for making the statewide management plan. Additionally, there was House Bill 821 that just passed out of the House and needs to go to the Senate. And that's maybe what she's referring to is this, you know, couple county effort where $500,000 for salt cedar, Russian olive, and buckthorn in the Yellowstone, Musselshell, and Stillwater watersheds. And so that would be for treatments and not defined yet. So I think that the task force has an opportunity there to guide and to coordinate some of that. And you're absolutely right, David, that, you know, $500,000 isn't going to treat it all anyways. So you have to go where it's best. And we always want to develop those other integrated management tools. So there's a science and advisory component to our planning grant. So happily to extend that invite to any of you that are interested in joining us for that conversation.
So, sounds great. Thank you.
Okay. So the one other thing that I wanted to bring up is Missoula County Department of Ecology and Extension will be moving into their new facility in August, and they have offered that we could potentially have our meeting there next fall. The fall is typically when we do our research updates. I know it's a long drive for a number of people, and we typically try and be closer to where the researchers are, but the building is shared with the insectarium and butterfly house. So there could be tours of a tropical butterfly house. It's a part of the meeting, might be a draw to get people to travel. We don't need to get people to commit. I just kind of wanted to put it out there as a possibility. And people can voice concerns or support to Jeff or myself as we move forward with planning that meeting. Really kind of varied the timing of when we do it. So I don't know if I would really tell anybody a specific date. It's been from October to I think January in the past. So there's some flexibility there too.
Sounds great.
I really appreciate the idea. I like it. No, we were saying it would be great if there was like a flight from Billings.
You can fly from Sydney to Billings and then jump in a car with us. Yeah, that would be a party wagon. We do have a minivan on its way. Oh, that's a lie, but you can fly in a minivan and then drive. Yeah.
Jeff, do you think it would be a good idea to take the next couple minutes to kind of talk about like some action items that came out of this meeting to just make sure we're kind of tracking some of those things that we discussed? Well, the action plan, maybe this handout idea, maybe other things that I wrote down I'm supposed to do, and you guys can tell me if I forgot anything.
I tracked some at the beginning
Okay,
and
that's
then
great.
fell off. One of them was entertaining the idea of a letter of support for an individual who is yet to be identified. Kenny's replacement.
Maybe that's the point, is like identifying someone who can do it.
Yeah, so I think the first step, if I understood correctly, was that I should send out an email to the email list, seeing if there's anybody else that's interested. And I might need some help on how to word this, because just because we're sending this out and we're looking for interest doesn't mean that we make that decision. It's just that we're choosing somebody to support.
Yeah.
I wonder too, if you, because I think Jasmine said she's sending out her
Yeah.
requests for people this week, and you could just use that as your base to
Okay.
share more information.
Okay, maybe, yeah, I'll wait until that comes out, so then I would have all of the details. Okay.
The info got posted on our website, but the PR will come out Thursday.
Perfect, thank you.
In terms of the action plan, just given some of the clarifying questions, it sounds like the breakout groups have a direction and they can continue to fine tune some of the things they brought up today.
Yeah.
And revisit it at a later date. But did you guys come up with a deadline? I thought you gave a functional deadline. I didn't capture that.
It wasn't like a solid deadline, but I think we said we were gonna meet with our smaller groups and kind of finalize the edits that we had discussed and hopefully compile those and get them to Lori before field season. So within a month.
We have a solid deadline.
Yeah, but I'm supposed to be ready for the deadline.
Yeah, maybe like our working group edits to Lori by May 1st. Is May 1st, like, are you guys already super busy?
That's a complicated question.
Okay.
Lori's gonna
Lori
be
is
busy.
actually taking on the job
Okay,
in Utah, Nevada.
yeah, okay,
And so she's got other obligations. So when you get it to her, she'll do it when she can.
okay,
That's-
which I
that's
think
the
is
answer to that.
totally reasonable, which we discussed, like.
But it's not
compiling it.
just her, because there's
No,
the group.
we're
I
on
also
there.
sent her a message earlier today and mentioned that if I needed to jump onto that group and help out.
Yeah,
I could.
Because I think we're fine with, like, if we can do something, that's great, but if we can't, pausing until September. Okay, yeah.
But try for a month. In our last meeting last fall, we kind of agreed to table, the discussion around featuring a spring meeting around pollinators. We were gonna talk about that this fall. Is that still something we want to revisit? Everyone's still interested in that. I don't have the exact language in the book for those.
Yeah, and I think, you remember Charlene, at our fall meeting spoke to continuing the pollinator conversation, and this is not something that we need to discuss in depth right now. But I'll probably need you at some point, Charlene, to help remind me exactly what we were gonna kind of, focus that conversation on, and if you had any speakers or topics in mind. So that would be our next spring panel conversation.
Those are the only action items I wrote down.
Okay, and then maybe potentially thinking about this handout
Yeah, if
that we
you,
discussed.
if you guys have the opportunity to look at that S400 stewardship guide and just look at that diagram where it breaks out pre-emergent and post-emergent tank mixes over the course of a 12-month period. I don't know, it's a neat diagram, and you can take that concept and apply it to biocontrol mixed with grazing mixed with reseeding, mixed with herbicide application, and it would have to be on a species basis. It's not gonna be a blanket, but it's an interesting idea, and if we can make it happen, I think there's a lot of great minds in this room that could add all that information into a nice pretty picture for us to use. So.
One conversation that we did have that didn't have a resolution, and I don't know if you want to revisit your biocontrol, your insect.
Yeah, the distribution
question.
I think, I mean, my kind of understanding of the takeaway was maybe it needs to be addressed with the weed coordinators at the fall meeting. I mean, people are usually very understanding, you know, maybe we don't have these like set limitations or guidelines, but I have conversations with every single person that requests insects. And so I think it's maybe possible to continue those conversations without having a set guideline this field season. If that's how everybody feels, like we really need to hold off until we get more solid feedback from more weed coordinators. The weed coordinators that were here didn't really have anything to say. So what was your thought, Joe? I'm gonna put you on the spot.
I don't really have any issues getting insects from you guys, and I don't ask for a lot, so
Yeah.
it hasn't been an issue for me.
And like we mentioned, there are very few people that typically request more than what we had on that document. And there's exceptions to that too, like we kind of heard from Brenda, which is a much different situation where she's working in, what was it, 14 counties?
Yeah, so
really
we could
significant.
Potentially incorporate in those limitations, like including geographical, like not reference, but like context. So like counties responsible for, etc.
Did anybody else have any conclusions from that conversation?
I think it would be really good to know just how many people this would be impacting and whether or not the story or some of those are more Brenda's story, or if it's more of, you know, shifting the conversation back to being targeted and intentional with the 20 releases or whatever that they're getting to help them establish. If there's really that need in that part of the state, then the focus is maybe more intentional on establishing insectaries there. They can, you know, nurture locally to meet those needs rather than. But I am concerned that maybe, you know, you wouldn't be moving this direction to have these side rails on. If you put that out there and people that even aren't really requesting that many insects have concerns about it, and it's not even impacting them. So.
Yeah. I mean, I think there was maybe 2 or 3 individuals. I just looked at flea beetles when Madeline and I were having this discussion, because that is typically where people request a ton of insects. It's usually flea beetles. And I think there was like 2 or 3 individuals that were over that 20. So it really is very limited. Yeah.
We re-message it a little bit and just say in the event that power and insects are limited, you may be restricted to this number.
Okay.
You know, and if we can accommodate them, we'll still accommodate them, but just let them know that there's a very good likelihood that you're gonna be restricted. So just maybe re-message it a little bit.
Okay,
Right,
I like
bring the
that.
message.
And then put it with what Bryce just said about, we encourage you to maybe start nurturing specific sites where you can eventually get your own.
Just increase that message so we're all jointly here to help them try to find out the places where they couldn't collect locally.
Yeah.
The turnover is so great in a lot of these places that they, you know, there may be places in the new people that are coming in here brand new, right? You probably don't know. And I know we've been up there, because she maybe Stone has good records, but, you know, we've gone up there and tried to identify where they're collectible. I know there are collectible populations. That's just an example, not picking on you.
No, it's fine.
As far as you know. Well, we're like this now.
I got a question about poison hemlock insects. Are those even available anymore?
Yeah, we got some, I worked with, some land managers in South Dakota to get some ships into Montana last year. So absolutely, like if that's something that you're interested in, I don't put those things on the survey just because there's not a ton of requests for them, but yeah, if things
We have
come up.
plenty of it.
I might be able to get you some too, Joe.
Yeah,
Okay.
and
It
Penny
depends
has
on
helped
the
me
year.
before too.
Depends on the year. On what-
I used to get them out of Bozeman from some outfit, but they quit messing with them due to the danger of the hemlock. So
Interesting. One of the commercial distributors was selling it. Okay.
Just collect the tanks.
Yeah, so let me know and I can work with you too. So we have a quarterly regional biocontrol coordinator call. And so all of the states kind of join up and this is a lot of what we talk about. If people are in need of something in one state and we can help, we help them and vice versa. So yeah, I'll just put that on my list here.
and we have a cost share for herbicide and biocontrol through our county. So
Okay.
if they needed to pay for the biocontrol, we could at least get them reimbursed.
Okay. Okay, I think it's good. I don't know if anybody else has anything they want to talk about. It's 4 minutes to 4, so. Okay.
Okay, to move or push to end the meeting? I'll move to end the meeting. Right, is there a second? Okay, meeting's over. Thanks everybody online.
Yeah, thanks, everybody.
Yeah.
Yeah, or you could just have, you're entitled to just have a nice holiday visit and not do anything else.
So I'm just hoping that's all she wants to.
Oh my God.
She said she wanted to explain herself.
Hold on, I have to scold my children. Liberty Rose, leave the puppy alone, put him down, her down. Sit at the table, both of you. Have a pencil in your hand and your homework on the table and stop fooling around. Or we're not gonna have any time to do fun stuff.
So anyways, so I'm sure it's gonna be all good and we're just gonna go and do Easter things and keep getting to know each other and just, building that bridge. I'm sure the time will come eventually where some of that's gonna have to come
I
up.
forgot to sign this.
But yeah. So okay, you go do those fun things with your girls or help them with their homework so you can do the fun things. I gotta do a few unfun things so we can go do the fun things too. So
Oh, you got a, what's that mean?
I built a
What is that?
touchdown is like a daddy tower.
Why is it making that sounds? Come here, we'll talk about it. So does that mean? Why is it doing that? Oh, hang on. Will you close my computer?